through your WhatsApp groups to come immediately, because people, if they are not talking, they are on WhatsApp. So you can easily reach to them. I didn't check my WhatsApp today. Tea was good? If yes, please raise your hand. Yeah, the tea energy. Tea or coffee? Who had both? There were some small biscuits, no? Not large, small biscuits were there, cookies. Where is Likit? I think we don't have time for any quiz or something. I thought 1 or 2 questions we can, but we require chocolates also for that. There are some last minute adjustments going on. We will be starting within a few seconds. Talking of tea, recently ICMR gave a report, right? You should not have tea after lunch. Why? Yeah, perfect. Tannins will prevent the iron absorption. And anemia is also a big problem in India. Iron deficiency anemia.
E is equal to MC square. E is equal to Einstein square. Energy is equal to molecular coffee.
Quiz, he's asking for quiz.
Quiz, huh? Okay. You take the chocolates. So, I will ask an interesting question. Let's see what the magic bag has got. I didn't expect such high demand, for the thing. Yeah. He's ready with the gold bars. No, we gave it to everybody. Okay. Okay, I'll ask a very simple question to begin with. Yeah? What is so simple? What is QRM? Yeah, that's what. We can't give chocolate for that. So I'll have to ask a more tougher question. Questions, Okay, there are 13 parts, right, for the new Schedule M. Can you tell which is the part related to computers? There is something related to computers. What is that related to in the 13 parts? What is it related to? No, that is a very easy answer. It is, okay, somewhat related to data management, but there is one more thing. I need something more specific. Sorry. Yes, sir. Please, welcome. Please give a big hand to Dr. Ishwar Reddy, sir. And it's a great pleasure for me to introduce Dr. Ishwar Reddy, sir. He is the Joint Drugs Controller of India. Please take your seat, sir, for just a minute. A very respected regulator across the globe and described as a thought leader on plethora of matters regarding drug regulations. As an accomplished professional for more than 25 years, he started his career in 1998 as the drugs inspector of CDSCO West Zone, Mumbai. And then he has done many workshops as well as audits on GMP, GCP, GLP. He was promoted to ADC grade of India in 2009 and appointed as Deputy Drugs Controller of India in 2012. And in 2014, he was appointed as the JDC, Joint Drugs Controller. A very industrious person, and as you have seen from morning, very sportsmanlike, always smiling. He is a very generous individual with respect to his leadership, so that makes him attractive to all people. And he has participated in several national, international seminars and workshops relating to drug regulations and undergone training from the very best, US FDA, KFDA, PMDA, WHO Geneva. He has been awarded with the Best Drugs Inspector Award in 2005, Distinguished Alumnus Award from Manipal Academy of Higher Education, Manipal. He also received the Best Drugs Controller Award in 2016 during the 66th IPC at Hyderabad. So he has worked as Drugs Controller General of India, and he is the youngest Drugs Controller General of India who has worked in various positions in different hierarchies. What a wonderful resume, sir. Before I feel jealous, I'll give the stage on to you. Thank you very much.
So once again, good morning to all of you. Thank you so much. So the first session, an introduction to product and process knowledge, mainly I focus about the regulatory perspective the subsequent speakers, they will go more in technical perspective. Because being a drug regulator, it is my responsibility to share with you what are our expectations and where it is a mandatory for you and how it should be done. So before I will go into the regulatory perspective, first, what are the issues, concerns, challenges at present we have? This is a most important slide. I think we need to understand about what are the issues, challenges in general we have. If you see in India, compared to the other countries, especially the big companies, in other countries, hardly in any one facility, single-digit products are manufactured. Or otherwise, mostly they are the dedicated facilities. But whereas in India, you name any company, and you name any dosage form, I'm not talking about 100%, by and large, you name any dosage form, all dosage forms are manufactured at the one given setup of the facility. And that's why this is a multi-product manufacturing facility. In addition to his own products, he is also having the loan license. And from this, his basket, he is not regularly producing all the products. I think, how many of you, raise your hands, you know how many product permissions your company is holding? Can you raise anybody? You know how many products you are holding, product permissions you have taken from the... Thank you. At least few are aware about that, the number of products. But I don't think so. He is saying number, but I don't know. List him, you name all your product permissions, what you have, whether he has remembered all his products or not. So what I'm trying to say that even when you are functioning in any organization, this is even same issue with the regulator also. Even if one regulator or that area inspector will visit the particular company, even inspector, I don't think so, he know how many products you are actually holding the license. This is one of the great concern for us. And I think that may be visual. Yeah, that is fine. So this is actually the problem. Why we need to have so many product permissions, out of which hardly, even if you ask me, 5% you are regularly manufacturing, 10% you may be occasionally manufacturing, and 20% rarest of rare whenever you get a tender for the government supply or for the export purpose. You are more than 70% of the products, hardly you see the light for that particular product. This is one of the major, why? Because of, we never had any product and process knowledge. That's why we never even remember what product permissions, how many product permissions we are holding. And can you raise how many of you in your organization you have formulation development facility? Oh, thank you. So this is also, if you see, hardly 10 to 15% of participants you have raised your hands. That means majority of the companies, even if you don't have the formulation development facilities, then from where you will get this product and process knowledge? That means you are producing the product directly by taking trial and error method. And we are taking calculated risk in that. So now, as per the revised Schedule M, you need to have documentary evidence about the product and process knowledge for each and every product what you are manufacturing. And under this, the three are the very important. One is the excipient compatibility studies. I don't know how many of, see, when I say that, I'm not questioning the entire industry. If you ask me frankly, 90% of the products, 85 to 90% of the products, they have a reasonably good data. Whatever products available in the market. Maybe a 10 to 15% of the products may be not having the data. When I'm talking about the 10 to 15% of the products. Okay, so these 10 to 15% of the products generally don't have any information about the excipient compatibility studies or stability studies. Then BA तो बहुत दूर की बात है. And you know how much resources are required to have the BA/BE data. This is one of the great challenge at present what we have with this 10 to 15% of the market. And the quality culture, I think we are going to have a separate workshop on the quality culture. This is one of the great challenge and we need to really address that. And continuous training. In any organization, training itself, they feel that, that particular human resources, their mandate time you are wasting on that. But actually training is the most important in our sector. Continual improvement, this is actually the first time I will tell you all, my dear friends here, the continual improvement word is specified in the revised schedule. Till now, this terminology was used in the ISO 9001, continual improvement. So this is actually another one session, what is the meaning of a continual improvement. But you should know that under the Schedule M also, it is the manufacturer's responsibility to ensure the continual improvement in the quality of the product. And finally, the data integrity. But when I said we have all these issues and challenges, but whether it is applicable to entire our sector, I say no. Why? These are the facts about the pharmaceutical industry. If you see, we as India provides the generic medicines to more than 200 countries. You name any country in the world, our products are there. That means our products are complying with their local regulatory requirements and also our products are more affordable compared with any other country supply. Because of these two reasons, we are able to supply more than 200 countries. And I'm happy to share you 10 out of 25 global generic manufacturers are from India. It is actually a very proud moment. That is the reason our first in the morning, we really appreciated ourselves by a standing ovation. So I have stated the top 10 generic manufacturers out of the 25 in the globe. And over 60% of exports to highly regulated market. So just link this data with the quality, with the compliance to the regulatory requirements. When I say 60%, what is the total exports approximately we have? So around $27.8 billion. That means in our currency, it is 2.25 lakh crore. I repeat, 2.25 lakh crore worth of medicines are exported. Out of 2.25, 60% of the export, that means almost 1.5 lakh crore worth of the drugs are exported to the highly regulated, like US FDA, RTZ, or PMDA, or the MHRA. If we don't have the process product knowledge, whether it is possible to do export? That means we have a knowledge, we have the facilities, we are able to comply. That is the reason if you see the number of ANDAs approved, it is 6,440 ANDAs till now Indian companies are holding. And we have 748 US FDA approved companies in India as of April 2024. And 90% of WHO pre-qualified are sourced from India. But from this data, as a Indian drug regulator, I just compare. If you see, to get 6,440 ANDAs, how many years we took? Maybe 3 decades. So we needed a 30 years time to get around 6,440 ANDAs. But compare this in India. How many product permissions every day state licensing authorities are granting? Maybe equal, around 6,000 product permissions every day. How it is possible? And for one ANDA, how much time it will take? Approximately? Approximately 5 to 7 years. Come on. So that depends upon how serious you are this thing. Even it may be a 10 months to 2 to 3 years if you do it on a priority basis. But how much time you take to get a product permission in India? Even the state authorities are delayed by 2 days. You ask for that. The government of that particular state or the CDSU is not functioning. They are not industry support. You put so many allegations. You wait for within 24 hours you want product permission in India. The same product permission, you wait for 10 months minimum. You generate all data for them. So now friends, earlier these are not the mandatory requirements for India, that's why you got the product permissions. Now whatever the 3 important product process related documents, Excipient compatibility studies, stability studies, as well as the BABE, these are the mandatory requirements. When you submit application for the product permission, first you should know whether you have the data or not. Stability studies, at least you need to have 6 months stability data. When regulatory authorities will ask you 6 months, you have a lot of excuses. But whether the same thing you can make a request for the US FDA to get the excuse? Why? I think now the time has come. So we have to respect our regulations also. So you need to develop, you need to have the facilities, you need to have adequate knowledge about the product and process. And if you see the overall market size, India is around $53 billion. So that is the reason I said 80 to 85% of the products what we have in the market, they have this data. But the only issue is the 15 to 20% of the products, which are lead to the 5% of the NSQ. So we need to fill that gap. Otherwise, if you don't address this 10 to 15% of the market size, it is going to tarnish the entire pharma industry image. You may be knowing recently some incidents about the cough syrups, how really it is badly affected our image. So that's the reason. So I appeal all of you, respect the Indian regulations also and comply to the Indian regulations as per whatever is prescribed. This, if you see, because of our quality, we have a product knowledge, we have a process knowledge, our exports are enhanced from the time to time. So recently it is almost $28.4 billion the last financial year our exports. And if you see the breakup of these exports, the maximum 73% of the exports are the formulations. And 17% is the API. And 4% value is vaccines and the rest is other products. So that means, so why I'm showing you this data is that this clearly indicating that as an Indian manufacturer, we have a knowledge, we have a capacity to develop any product, not only a formulation, even API or even intermediate. We have very good knowledge. Just you give the powder, one white powder to a manufacturer, he will, within 24 hours, they will produce the product also. We have so much of knowledge, especially the chemistry. And even we have so much of knowledge in the formulation. We have good experts in the tablets or any other dosage form. If you don't have that, otherwise how we can achieve this? With this, we are able to address the market of 85%. The rest 15% is a great challenge. So that is the reason we are here to even comply the 15% also of about the requirements. So what is the actually objective of this workshop? Why we are conducting is to provide insight to all manufacturers about the product and process knowledge, and to understand the regulators' expectations, and to disseminate the new provisions to all stakeholders, and also to make manufacturers ready for implementation. Because as Mudda Sahab said, these now couple of weeks are remaining for the major companies, and the couple of months are remaining for the MSME for the implementation. And what at the end of the day we'll expect from this workshop is that you are going to be actively participate in the workshop, and this is going to be significant awareness about the new provisions, and I'm sure definitely it will upgrade your knowledge and it will help in the ease of administration. Why actually product process knowledge is important? Why can't you directly produce the commercial batch? What generally some of the companies may be doing. But what is the risk associated with that? If you produce the product without having any knowledge about the product and process, as you are aware, in pharmaceutical industry, we are not testing each and every individual unit before it is released into the market. Individual means each tablet, each vial or each capsule. Are we testing each tablet capsule? Because of our testing is a destructive testing. We test only random sample. If you produce 1 lakh tablets, out of that you test 100 or 200 tablets. 100, 200 tablets passes, remaining tablets you release into the market thinking that that tablets also will comply with the requirements. But unless, if you have the data, if you are sure the remaining tablets also passes, whether with respect to the consistency in the quality or the uniformity of the strength and everything, that is only possible if we have this knowledge about the product and process. And another very important, consumer don't have access to the quality, and he is not able to assess the quality. At least when you purchase any mobile, you can assess the quality of the mobile, what you are purchasing, and you also have an option, which brand of mobile you can purchase. Can medicines patient have any option? I want only this particular brand of the medicine. So that means he has to, whatever the doctor prescribed, he has to purchase, and he has to trust that particular brand, whether it is generic or brand. And in case of medicines, low and high dose both have an impact. It is SNI of 15% in extra we will give, or 20% extra, 20% discount we will give. This type of marketing does not work in the pharma. And NSQ is one of the major issues because of lack of the product and process knowledge. And para 1.5 clause B of Schedule M makes a mandatory for the compliance to the product and process knowledge at all stages of the product cycle. And it is, PQS clearly says that manufacturer must assume the responsibility of the quality. These are the first time in the regulations it is stated. And also fix the responsibility at all the levels. Even senior management has the ultimate responsibility. And the fourth point is very important. You all come under the fourth point. So you should not tomorrow say that, I am not aware, I am the shop floor, I am the only chemist only. Okay, I am the only QA manager or the maintenance manager or whatever, irrespective of your designation. Schedule M fix your responsibility also under the fourth point. That is participation, your participation. Support, your commitment of staff at all levels in all departments. You read the Schedule M very carefully, the PQS. That means your participation, your support, and your commitment towards the quality of the product you are producing is also very, very important. That means tomorrow you may not be able to raise your hands. I am not responsible for that. So it is a combined work. Everybody is responsible to ensure the quality. And another very important, as Mudda Sahab also said in the morning, first time under the PQS it is stated product design and development is a part of quality management. Earlier these terminologies are not under our regulation, but first time we have incorporated this. And also the first time the regulation stated consistency in the quality. So when the inspector come and ask you how you are ensuring the consistency in the quality, what reply you give? Unless if you have your process and product knowledge, and that is formulation development information. And what is the basic fundamental principle of the product and process knowledge? The basic fundamental principle is that the quality should be built into the product. Yeah, this is the slogan for everybody. Till now, some of the industry think that quality means you test the final product. If it is complying with the given standard specification, that product complies with the quality. So quality should not be assured by merely testing the quality of the product, but your quality should be built into the product. How? With an understanding of the product and process by which it is developed and manufactured. You should understand. And along with the knowledge of the risk involved in the manufacturing of the product. That's why we have a separate requirement of QRM. And finally, it also says how best to mitigate those risks. How you are able to reduce the risk associated with respect to the quality. If you don't have this formulation data, what happens? Like recent incidences with solvents in cough syrups. In fact, it was so surprising, even during the audit of the cough syrup manufacturing facilities, we observed even some of the manufacturers, they have not tested the solvent at all. Vendor validation and other thing is the secondary thing now. Now even so that particular manufacturer, even they have not tested the solvent before used. See how much risk is involved. Why I'm telling you these examples is, one side even we have such a manufacturers, even they are not testing the even incoming raw materials before they are using. And another side we are talking about the product process knowledge, quality, design and development, continual improvement. So what is the gap? You understand what I'm trying to say? So we have to bridge this gap. We have to bring these people also to on par with the standards. The main objective of this type of workshop is this: fill that gap. I know many of companies may be doing very well. Okay, you know much better than with any other. But only thing is that wherever the gap is there with some of the people, we have to build that gap. And second, if you see, I think you may be knowing CDSCO will post monthly the NSQs on our website. So an average 1500 to 2000 product samples data we will provide. Almost around 4 to 5% NSQs we observe. If you see the NSQ, majority of the products are failure in the dissolution. Because the quality of the excipients used, even the technology, even the manufacturer is not aware. They are using such a low-grade excipient. Majority of the dissolution and some of the products are failed in the assay. Reason is that manufacturer is adding the sufficient quantity, but the product is not stable, or the product is not having the consistency in the quality in each dosage form. These are the inbuilt problems associated with the product quality. And unfortunately repeated failures. And a very interesting thing is that whenever generally, whatever I observed, whenever your product fails, most of the manufacturers claims only one reason. This failure because of the improper storage at the retail, wholesale, at the distribution channel. This is a evergreen reply. You name, by and large, most of the companies, okay, so this is not properly stored at the distribution network. then who is the responsible for that? Manufacturer or regulator? If it is not properly stored, distributor is responsible. Who has authorized the distributor by the manufacturer? Whether manufacturer is authorized or not, or the drug regulator is authorized. Drug regulator has given only the approval, a license to stock, to sell, or to further distribute. But this is the time has come. Why I am correlating this? This time has come whenever your product is NSQ. Now inspector will ask you the complete root cause analysis report. Even if you see the revised schedule M, it is your responsibility to give the complete root cause analysis. That means even if you want to claim that particular retailer, wholesaler, he has not stored properly, you have to prove that particular facility he has not maintained the temperature. That means you have to generate the data. You have to do temperature monitoring of his facility and to show data that yes, I have monitored this distributor for 1 month or 1 year. His temperature is not proper. That's why my product is failed. That is the main. You are really prove that that product is not properly stored. Otherwise, we are just making the blame on the distributor. Do you have any documentary evidence he has not stored properly? And have you done any audit of the distributor, or have you prepared any report? So, what is the point for discussion here is that whenever your product is failed, you had to conduct analysis, including you go back and review your complete formulation data, development data also. Is there any problem with the formulation development itself? How many of you really changed your formulation after NSQ is found? So NSQ, you just do maximum, you give always 3 standard reply. One is that we have done the testing of our control sample by in-house, by outside laboratory also. It is found standard. That is a and similar 2-3 other batches also of the same product we have tested. Product is complying and the storage condition. Now time has come, we have to go beyond that. You have to go and see really the root cause for the NSQ of that particular product. And NSQ unfortunately more with the government supply. You may be knowing why it is more. 2 reasons. One is, whenever you participate in the government tenders, that product you never manufactured. First you get the tender, then you start production of that product. That means where you have the product process knowledge. That means without having the product process knowledge, you are supplying to the government. I am not saying that all supply by all the manufacturers. I am saying the specific. That is whatever I am talking is the 10-15% of the supply chain. So without having that, they will supply. And similarly, export to the unregulated market. This is a very high risk involved in that. Because of exports also, you don't know which product you get the order. That's why you don't have the data for that all the products. And first you get the export order, then you go for the state licensing authority to get the product permission, and then you export. And where you have time to develop that product. And that export order, you take the commitment in 2 months, 3 months, you have taken commitment to supply. And within 2-3 months, your procurement of raw material, packing material, other things so itself will goes. Then where and how you have done the formulation development of that particular product. That means without having any knowledge, we manufactured and exported, especially to the unregulated market. So there is a very high risk involved in that. And because of this lack of formulation data, there is no much trust and confidence on generic medicines of certain manufacturers. But if you have a formulation data completely, what are the advantages? What actually you assure? So you can, it is actually the main, whatever the purpose that product you have developed, it will be suitable for that intended use, and there will be a consistency in the quality, and especially the dose uniformity, especially when you have narrow therapeutic index drugs. And it should be stable over the shelf life. And it should be compatible with the excipients, and this bioequivalent with the reference product, and your all process are validated, you have the process knowledge, and very important, you are complying to the regulatory requirements. This is the most important. So having the formulation data, you are going to be assuring that. These are the, why I said, these are the mandatory requirements under the regulation. That's why what we are expecting from you. You don't have any option. If you see the BAB studies data, as per the government notification dated 3rd April 2017, you had to generate the BAB data as per the biopharmaceutical classification system for class 2 and class 4 drugs. In the subsequent slides, I will explain to you what is 2 and 4. For grant of license of oral dosage forms. And similarly, stability, excipient compatibility data is also mandatory. And even Rule 71, 76, 76CS says that the drug shall be safe for the use in context of vehicle, excipient, additives, pharmaceutical aids. That safety of that particular excipient, what you are using, also to be assured. And as I said just now, I said product design and development is a mandatory under the para 1.3 of Schedule M. And the process and product knowledge, as already we discussed. But if you see, all this data is a mandatory for you to get a product permission. It is not that, first you get the product permission, then you generate the data. While you are going for any particular product permission, whatever the reasons you want to have that product permission for export or domestic or for the government supply, first you need to generate this data and submit to the regulatory authorities for getting the product permission. And regulations also clearly says to generate this data, first you need to have the Form 29 license. You have to submit the application in Form 30. I think most of you may be aware about this called test license, Form 29. Without test license, whatever the data you generate, generally regulatory authorities are not going to be considered. Okay, so that's why to manufacture any drug, small quantities for the purpose of examination, test analysis, and these batches are manufactured for the generating the CMC data, conducting the stability studies and bioequivalence, and this data generated for regulatory submissions. And the product, what you manufactured under the test license for generating the CMC data, stability, other thing, it is not for the commercial use. And you may be aware how this license is granted. You have to make application. Sometimes state authorities will go for inspection if it is required. Then you have to obtain the license. Then you have to generate the data and submit to the SLA for getting the product permission under the suitable license form. So from this slide, what message we are conveyed to is that whenever you are generating the data for regulatory submission, whether it is a stability data, BABE, or whether it is a excipient related compatibility data, first you get a Form 29. Otherwise, after generating the data, if you submit, the data is not considered for the acceptance for any approval. And these are the general considerations in the dosage form design. Mainly it talks about the first you need to do the characteristics of the drug substance, and you need to conduct the drug stability. You should determine the drug formulation stability and prevention against any microbial contamination, separation and palatability, therapeutic considerations in dosage form design. This is the, I think the subsequent speakers talk more about all this. Generally, I will, how much time I have? How much time I have? 10 minutes. Oh, okay, then I need to do 2020. So let me see, I think all of you know the particle size, how particle size is going to affect the bioavailability or the dissolution profile and stability. And we need to understand the particle size properly, and polymorphism also plays a very important role in the design and development of the product. I'm skipping that. Solubility is also, And if you see the certain products which have the narrow therapeutic index, see for example products like misoprostol or levothyroxine, it is a 0.1 milligram. How you assure the consistency in the quality, and each and every tablet if you test, it should have a 0.1 milligram. Unless you develop the process, unless you do the validation of the process, then only you are able to assure that. These are actually the sequence of developing the formulation development. You would start with the literature survey and active drug substance, active product. I think I can skip this. Anyhow, we are going to share the slides with you. These slides talks about how you should really do your design and development. And this is the very important, safety of the excipients. This is which is a mandatory requirement. Okay, excipients plays very, very important role. And if the excipients are already, some precedence is there for use, and you no need to generate much data. And if the excipient is completely a new excipient, you have to do lot of safety data, you have to generate. And there are separate guidelines on that, what type of data need to be generated for the new excipients. And stability studies, I think all of you know very well. Actually, this is the one of the major reason why the 5% NSQ is in India. Because of we never conducted this stability study for that particular products. Okay, which is a mandatory requirement as per the GSR 630 from 2018 onwards. And you had to submit the evidence of stability to the state authorities before getting the product permission. And mainly, as you know, the stability study data is generated to assign the shelf life of the product. And while you are generating the stability study, the container, closure system, packaging, also keep it in mind, actually which container you are finally placing in the market. Your stability study has to be conducted as per that particular container. And actually during the stability studies, what you test, that selection of testing parameters is very important. You have to do some stability indicating parameters. I am not going much details with respect to this, but at least minimum 6 months stability data, both accelerated and the real-time data you need to generate before asking for any product permission. You have to do analytical method validation that used for the stability purpose. And I'll take a little more time here because this is one of the new requirement, and you also maybe not much implemented this. This is the most important point. Because of lack of this data, that is the reason we don't have we means in a public, don't have a trust confidence on generic drugs available in India. But generic drugs which are exported to the highly regulated markets, they are having the global standard. But the generic drugs which are supplied in India, main reason, again I am telling you, 100% generic medicine, 15 to 20% generic medicines which are available without having the complete information. So I think you may be knowing what is the bioavailability. Bioavailability is mainly the rate and extent to which the drug is absorbed and become available into the systemic circulation. Okay. So if you see this graph, I mean, you may be knowing how the BAB data is generated, the particular dosage form, tablet or some capsule administered. But very important thing you observed in my slide also, I said, BAB data in India, it is required only for the oral dosage form. Okay, not for the other dosage form. But some other countries, even certain categories of injectables also require a BAB data. But fortunately, I don't know why, but if you see Indian regulation talks about BAB data for only oral solid dosage form. Okay. So the data is generated, so the tablets are administered to the group of the healthy subjects, and at quite frequent time they will draw the blood sample from the particular subject, and they will estimate amount of drug present in the blood, and then they will plot the graph. At 0 time of blood sample, how much drug was present, after 1 hour, 2 hours, 3 hours, there is a different frequency has been defined, and you plot the graph with respect to the time what was the plasma concentration levels. And if you plot this graph, and if you see the curve under the area under the curve, which actually represents the total amount of drug absorbed into the systemic circulation. Is it clear? And the problem with and for the bioequivalence purpose, what is actually the generic product? But unfortunately, in our entire regulations in India, there is no definition of generic drug. Either you see Drugs and Cosmetic Act, drug rules, or any regulations, even the guidelines made by the regulator, nowhere it is defined what is actually a generic drug. There are definitions of generic drug outside India by different regulatory authorities, but Indian perspective, there is no definition. So this generic drug means the drug product which is identical or bioequivalence to brand or reference drug with respect to the active ingredient, same route of administration, same dosage form, same strength, same indication, and safety also remains same. But may be different in active ingredients, may be different in color and shape. If the subject drug is bioequivalent with the innovator product, then this particular, the first graph, the original and generic is almost coincide with the graph. This is the bioequivalent, that product we can say. But whereas second one, if you see, one particular innovator product and the generic product, it is not at all correlated. It is not superimpose each other. So that they are not bioequivalent. But the actual problem is, this is the problem. Means, if you see, this is for example, the first peak is the innovator peak if you take, to have any drug effective, as all of you know, the minimum concentration in the blood is required. Do you agree that? When for example, take paracetamol tablets. So the paracetamol, once it is drug enter into the stomach, it will get disintegrate, it will get dissolved, then it started absorbing into the systemic circulation. But immediately after absorption, we don't get the effect. But we need minimum amount of drug should be in the blood to get the efficacy. So that is called the minimum concentration level. If you see the innovator product, how much time the plasma concentration levels are maintained. But if you see the other products, some is a only short time, and other one particular generic, even it is not achieved never reached the minimum therapeutic concentration level. But all three graphs, all three products, area under the curve is remain same. That means the amount of drug absorbed into systemic circulation is remained same. It means even if I will draw the sample from the market of the generic drug, I will analyze in the laboratory quality control test if I will do, it passes with the test of assay. Assay is having 500 milligram. Innovator is also 500 milligram, the generic drug also 500 milligrams. But the generic drug, that particular product, that is never reached the minimum therapeutic concentration. That means it is slowly disintegrated, slowly dissolved, and absorption taken long time. It is never achieved the therapeutic concentration. The main problem with the generic drugs, what we have certain where you don't have the product and process knowledge, it is going. That is the reason if you see in India, not only India, globally, only certain companies, certain particular product, it moves so well. Why not other products also? The same different companies can move the same thing. You make any one, for example, metformin tablets. For metformin tablets of one particular manufacturer, it goes so well because of their product is more having the bioequivalence. Otherwise, many products of many companies are producing the metformin, but all are not that selling well. They may be having the same amount of 500 milligram of metformin, but the amount of drug really entered into the systemic circulation as per the innovator, that is really an issue. So as per the biopharmaceutical classification system, the products classified into 4 categories: highly soluble, highly permeable. Mainly class 2 and 3 are important. Why? Because of they are the low solubility. I have given the meaning of what is the high soluble and high permeability. So the BAB studies are required only for the products comes under the class 2 and class 4. Because of these two categories are the low solubility. And you maybe have doubt which reference product should be used as a comparator for doing the BAB study of the your product. Either identified by the regulatory authorities as designated as a reference product, or usually the global innovator product, or any formulation approved by the DCGI after full evaluation. And I think due to lack of time, I'll skip this. Generally parallel designs they will do, either two-group parallel design. One group they will get reference and the other test, and otherwise we have crossover design. After washout again, the test and reference given to the two groups.
You can go ahead, no problem. 5-10 minutes is not going to make a difference, we'll have some more on.
Okay. So because these studies are the very, very important, because of till now, you are not involved in that. Okay. And these are also expensive. I mean, at least minimum 12 lakhs and above it will cost the study. Okay. And you maybe also see that how many subjects that study should be conducted, and whether study should be conducted on healthy subjects or study should be conducted on the patients. If you see, generally BAB studies are conducted on the healthy adult volunteers. Okay. But however, there are certain drugs, there is a risk of toxicity. See, for example, cancer drugs. So we may not be able to do BAB studies of cancer drugs on healthy subjects. I think you know very well. There is a lot of risk associated. So in that case, the BAB studies are conducted on cancer patients itself. Okay, that is the one point. And generally for you do the pilot BA study, as per our guidelines, the minimum number of subjects shall not be less than 16 subjects. And we have, CDSCO has given the guidelines to conduct the BAB study. And also the sample size depends upon either inter-subject coefficient variability or the test reference ratio, and how much power you are giving to your study, whether it is 80% or 90%. As the more power, the sample size will increase. And also what is the significance levels. So, if you see the regulatory considerations with respect to the pre-formulation, these 4 are very, very important. One is the quality related. So we need to have the pre-formulation data which supports the implementation quality by design. See, in my entire presentation, I never use the word quality by design. But indirectly, whatever our regulation does not, talk about quality, we dare not use the word quality by design. But indirectly it attracts the end of the day quality by design. So that's why subsequent speakers, they talk about the quality by design. But what you should, for you important is that how the quality by design compliance should be made in your facility to achieve our Indian regulatory requirements. And another is the safety. Through pre-formulation studies provide the data on the safety profile of the drug candidate, and efficacy is of course, and finally, which is also very important for the compliance perspective. So to conclude my session, my topic on process and product knowledge and overview. So I appeal all of you is that whenever, if you don't have the facilities for the formulation development, as I said in the morning, kindly you had to set up your facilities. And also I said in the morning, if you are not able to have so much of resources to set up, you need to tie up with suitable outside agency. It may be academic institutes. It may be, for example, NIPERS. You can tie up with NIPERS. So NIPERS on your behalf, they develop formulation and they will provide the complete details to you. Or also you can purchase the technology, the dossier for you. So it is regulation not only restrict that the data you only have to generate, even the data which is generated by other company, provided if they will give the data to you, okay, that data is also acceptable, but that is through your technology transfer. You need to do the minimum studies to ensure the compliance to the technology transfer. And as I said, it is a mandatory to submit the excipient compatibility data, stability data, BAB data, and thereby definitely it will reduce the NSQ, product recalls and product failure. And it will also promote the exports. If you have a complete data, knowledge about the product, and you have full confidence on quality of the product what you are manufactured, thereby you are ready to export to any other country. At present, you may be having some reservations about the exports. And if you have the complete data, we are going to be comply on par with the global standards. It will also build the trust and confidence on quality of the generic medicines. And main takeaway message from this session is that, as I said in the beginning, your role, you have very important role as per the revised schedule. So your participation, your support, your commitment, whatever the division you are, to ensure the quality of the product is the most ultimate goal. So you have a lot of important role in assuring the quality. Thereby really you are taking care about the public health end of the day. And whatever you associated with any company where you are producing your product, these products are taken by your family members, your friends, or your any other relatives. So first of all, as a manufacturer, you need to have a full trust, confidence on quality of the product you are manufacturing. And again, I am saying you here, quality means how you define the quality is the most important. Testing the only final product and compliance to that testing report to the given standards is not the quality. That's why the basic principle of the revised schedule, the quality should be built into the product. If you understand the meaning of quality should be built into the product, if you ask me frankly, if the quality is built into the product means we no need to have testing, we no need to test the finished product. But I am sure the days will come in our regulation, there won't be any quality control laboratory in pharma industry. Don't worry, those who are working in quality control department, you lose your job. What I am trying to say that the quality is assured at the each and every stage itself. Either it is at process or by using artificial intelligence or machine learning. Already we are in the industry 4.0 generation. There is a lot of automation is there. Even BMRs are, e-BMRs are available. The complete artificial intelligence, machine learning, you name any technology, almost for my majority of the industry, those who are involved in the highly regulated market, they are using. So once the quality is built into the product, if you are confident on that, then why you need to evaluate the final product in the quality control laboratory? And this is like this, in any classroom, in a college or school, if the master is perfect, school is perfect, systems, procedures are there, वो student exam लिखने की जरूरत ही नहीं है. You understand my logic? आप इतना अच्छा पढ़ाई किया, okay, then what is the objective of writing the exam? You are writing exam means somewhere in the system is wrong. Whether school standards are not good, our master is not good, or otherwise our syllabus is not good, or otherwise combination of all. So unless we strengthen our system, that is what I am trying to say, that the quality should be built into the product. Then we no need to worry about the quality control and any other, even not only quality control your side, and Indian regulators also no need to draw the samples in the market. If you see in USA inspectors, they don't draw the randomly samples from the market without any reason, unless if they have a complaint in the quality, unless they have any suspicion. But in India, we are assuring the quality by drawing the sample from the market. So drawing sample from the market means we are doing only quality control test. So chemically it may be a passing, but therapeutically it may fail. I shown you in the graph. इसमें paracetamol 500 milligram chemically if you test it will be present, but out of 500 milligram tablet, how much amount of paracetamol really entered into the systemic circulation? That is the most important. So to assure that quality and compliance and ensure the basic principle of the quality should be built into the product, we need to have a process and product knowledge. Thank you. Thank you very much for your kind attention. So I think lack of time.
Thank you very much, Dr. Ishwar Reddy, sir. Truly, we require that data mindset. And you gave a very illuminating point on the BA and BE point. So, you know, in marketing, we used to say, doctor, chemically equivalent drugs doesn't mean therapeutically equivalent. So that reminded me of that. I request Mr. Sunil Attaur, sir, to kindly offer a bouquet as a mark of respect and thanks for offering this fantastic talk on introduction to process and product knowledge. We'll take the Q&A at the last, that is at the end of the day. And surely you will have a lot of questions. So we'll proceed to the next speaker, none other than Mr. Harish Jain. I request you to please come over to the dais. Mr. Harish Jain, National President, FOPE, and Director, M-Biotic Laboratories. He will be deliberating on formulation development by quality by design. So he takes off from where Mr. Dr. Ishwar Reddy sir talked about. So the next level is QBD. He will be touching on various points such as literature search, pre-formulation studies, compatibility studies, and so on. Please come, sir. You take your seat. So Harish Jain sir is BPharm 1989 and MPharm 1991 from Pharmaceutics from the College of Pharmacy, New Delhi, which is now called DIPSR. And I think he requires a round of applause now for his GATE score. First, understand. GATE score is 99.74 percentile. So you can imagine he did very well. So he's a pharma entrepreneur with 31 years of experience. He's the national president of FOPE, served for 10 years as treasurer, secretary, president of KDPMA, elected member of the Committee of Administration at Pharmexil, chairman, Pharmexil Regulatory Committee, member CIA, National Committee on Pharmaceuticals, governing council member of LSSDC, member of expert working group of Indian Pharmacopoeia Commission, vice chairman of CIPI, Confederation of Indian Pharmaceutical Industry, effectively represented FOPE and KDPMA in different state and national forums, committees with respect to the small-scale industries and various issues, including such as hand sanitizers in Schedule K of Drugs and Cosmetics Act. He is also the past member of Vision Group of Pharmaceuticals, Minimum Wages Advisory Board of Government of Karnataka, past member of Sub-Therapeutic Committee of the Karnataka Drugs Logistics and Warehousing Society, and Joint Secretary of Organizing Committee of Golden Jubilee Celebrations at GCP Bangalore, Co-Chairman Exhibition Committee IC Bangalore, Regional Coordinator 68th IPC Vizag, recipient of Scotch Achievers Order of Merit 2014, Certified Corporate Director, Media Panelist with Sunset TV, and a lot more things are there, associations with the academia such as MIT University Noida, the CGMP Center at Mahe Manipal, that is MCOPS, then also with the Business Management and Consulting Committee and Company at Shanghai. He has received the guest of honor and delivered the keynote address at the World Congress in Pharmaceutical Sciences. He has successfully conducted workshops. So, sir, the impressive biodata, we are awaiting your impressive talk. The stage is yours. Thank you.
Thank you, Sunil. I think he forgot one important aspect. My blood group is O positive. Thank you, Sunil. I think much more than what I deserve. And I think before I start my talk, I thank CDSCA for giving me an opportunity to speak on a subject which is very dear to my heart. And I'm speaking in front of some of the top regulators in this country, the top scientists in the country, my friends, my seniors. And I invite a critical review and criticism for what talk I am going to deliver. Now, on a technical subject on this forum like this, I'm delivering my talk for almost for the first time thing. Otherwise, Otherwise, for almost more than a decade, being associated with various capacities in KDPMA, I've almost always talked on policy advocacy, regulatories, and trade matters. But then I'm happy that going back to where actually I wanted to start, a subject which is very dear to our heart. And now, coming to the revised Schedule M, as we are talking about revised Schedule M, as Dr. Ishwar Reddy ji has set the tone, and I think many of the things which I wanted to say he has already covered, so that makes my talk much more easier, and already that context is set. Now, as we are talking about Schedule M, apart from as far as product development and everything is concerned, there are certain points are there in the revised Schedule M as well. But the more details are in the guidance document. I think this is where the reference I have taken from, the guidance document which was issued by DCJ's office a couple of months back, which requires every applicant should submit the kind of data for obtaining a license. Now, this is where I have taken that base of my talk, because I thought that will be much more appropriate. Now, if you look at what is happening in last 1 year's time, the Indian regulators, the Indian government want all of us to upgrade in terms of quality, in terms of our compliances. Is there any escape? Unfortunately, no. Unless we upgrade our systems, I'm not differentiating here whether we are small, medium, or a large companies. I can give you examples of many large companies who are not non-compliant, and there are, on the other side, small companies are over-compliant. So let us not go into that aspect, whether it's a small, medium, or a thing. If you see most of the top 20 companies in this country today who are doing like 20,000, 30,000 crore turnover, and so they are into the top 50 in the world, all started small. So I can give you so many examples. So whether you are small, medium, or big, we need to have, if you want to stay in a medicine business, you want to stay in a pharmaceutical business, quality is must. There is no compromise in the quality. And as Dr. Raghuvanshi ji always say, you make a product which you can give to your sons and daughters. So that should be the kind of quality and the trust you should build into the product. So today when we are talking about building the trust and the quality at every stage of the product, I also say when you're developing a product, you should build the quality at every developmental stage. And this is what my talk is all about. Now, all the Indian government as well the regulators is preparing us for the future. When we talk about $425 billion of business by 2047, that's what Modi, our target is, not that $120 billion by 2030. Our aim is $425 billion pharmaceutical markets. And it's not going to help Indian consumers, but also the patients all across the world, because it's only the India can do it. And there's a big, huge opportunity for us. The government is preparing us to leapfrog in terms of quality and value. Today we always talk about India is the pharmacy of the world. We are the largest generic manufacturer. But are we touching the bulk of the market? I am afraid it's not. The huge business lies with the innovative dosage forms. The huge business lies with the new chemical entities. The huge business lies with the new biological entities. So initially when we when they are making us ready in terms of the generic development, generic quality, and reducing an NSQ, they are preparing ourselves for the bigger picture. Can't we go to, do we have to travel to that level? Yes, the answer is yes. This is where my talk will be focusing on. Basically, it's on the development on the generic products. What are the various steps are involved? Now, I just wanted to show you this documents, which was released guidance document for submission of application for obtaining manufacturing license by the applicant based on the dossier-based licensing of drugs. Now, this document has two parts. Part A, which you can see, which is related to your company's more of a KYC. The second part is the product-specific details. This is where is important when we are talking about the formulation development. Now, what are the important requirements of these guidelines as per the Part B? We are talking about process development report with excipient compatibility and force degradation study. It's again, it's also there in the part of the Schedule M, as well as the, I think most of the scientists who are working day and night for almost their whole life, they have spent on formulation development. For them, it's not a new thing. But then today, it is a mandatory requirement. We are also looking at process validation report. I think my good friend Prashant will take that in more detail later. We're talking about analytical method validation report, stability studies data, comparative dissolution profile, BE protocol and report. So these are some of the mandatory requirements which you are supposed to have. Now what I have done is, while discussing about formulation development, I have taken mostly tablets as what should be our strategy for development, because there are certain reasons are there. I'll come back to come to that later. But as an example, I'm taking tablets as the for the design strategy what needs to be developed. Now, development of a new drug product is always, I say, it's like we need to design a product even before you start thinking about developing a product. In our mind, which obviously has to be put in paper, what should my product be looking like? Correct? When we go to a tailor and when say, okay, you stitch my dress, what should we say? You should have one top pocket on the shirt. It should be full sleeves. It should have so many buttons. Okay, I'll give you a cloth which will make a shirt, but then it ends up making a jubba. So this is what we case. Even before you start developing a product, you should have, when you say about quality by design, you should know and visualize what should my product is, what are the critical attributes of my product, finished product, how it will physically look like, what should be a dissolution rate, what should be its bioequivalence profile, and what should be the size of the product. Many of us, especially many of us, when we started, what we do is, this is going to be my final product cost, and then we design the product. No, that doesn't work like that. That's what we talked about. We should be looking at defined release specifications, ability to maintain physical, chemical, microbiological properties, such as description, hardness, friability, dissolution and disintegration, impurity profile, and stability within the acceptable limits. These are some of the mandatory requirements are there till the end of your shelf life. Now today we are talking about quality, not only the quality of the product when we release from the factory, or it is at the retailer level. Today we are talking about quality of the product in the hands of the patient till he consumes. This is what we are talking about quality. Should comply, our development strategy should comply with the principles of consistent quality. We always talk about what is our whole business lies on quality, safety, efficacy. Now, without these three principles, can our business survive? Absolutely no. And I would add another 2 points, trust and compliance. Now, all this depends on meticulous planning and meticulous development work till it is launched to the product. It should be, as I said earlier, even your development work should be from day one and every stage. It's not be that you do the development and after 6 months your stability fails. It's all loss of time, loss of money. The purpose of pharmaceutical development is, it's basically when you're looking at a drug product, we are looking at basically 4 different aspects. We have a API, we have a excipient, we have a manufacturing process, and effective packaging. So our own product is basically composed of these 4 parameters. Now all these 4 parameters should be of a defined quality, and the process has to be robust. And development process has to be robust and suitable for intended use. You can't make a product which will not be able to use it. Now when we are looking at what are the critical factors, risk strategy and statistical evaluation. Every step you should have is risk management strategy. And even that without this, when I talk about various strategies and later on in my presentation, how a risk management plays a very important role in your whole development process. As I said, why I am talking about tablets. It's a kind of a universal dosage forms. There is no person on this earth who has not taken tablets. Not only in the allopathic system of medicine, we are also talking about whether you take homeopathic medicine, whether you are taking about Ayurvedic, nutraceuticals. So it's a most common dose form. But tablet manufacturing also involves too many processes, too many variables. Although it looks very simple, a small tablet, a white colored crocin tablets, 600 milligram 500 mg tablet, but then it looks so simple, but then so much of variables. I can tell you even, I don't know how many billions and billions of trillions of tablets, paracetamol tablets are being made every month in this world. But still, no one can say that mine is, only NSQ is 0. Impossible. This is because the whole thing is that the process variables are so much, but then how do we minimize? Now, when you talk about development, I listed out what are the sequential. I'll just run through that. We're looking at theory and review of literature. Later on, maybe in my presentation, I'll take few of them in much more in detail. We are talking about preformulation studies, stability of API and excipient, solid state stability, temperature, water content, and hygroscopicity, mechanical strength. Very important, API, excipient screening and compatibility. How do you do that? Risk management, as I said. API, very, very critical. How do you select an API and how do you select the vendors for the API? We should also need to study what is the innovator's product. Patent landscape, very extremely important. I'll tell you some of what are the steps are involved. API source identification and evaluation. When I say about API source evaluation in the development stage, it's also important that we as a smaller companies, how can we select or we can do a vendor validation in very simpler way. Today, vendor validation is one of the most important thing, as Dr. Ishwar Reddy ji has talked about, but then most of us fail to do because of practical problem. But how can we do sitting at our own office? How can we do some kind of a minimum vendor validation has to be done? But yeah, somewhere you make a start. Later on, we start with first standard, second standard. Later on, after 10 years, we will complete SSLC and graduation. But let us make a start. If you don't join the school, what is the point? Container closure and packaging system, manufacturing process, analytical evaluation, process optimization, establishing in vitro, in vivo correlation, scale up, process qualification, pilot batch, bioequivalence study, development audit, validation batches, commercial revalidation batches. Now with this, so much of steps involved, and I'll tell you none of these steps can be compromised. Now, Dr. Ishwar Reddy ji talked about every company we can have around 10 products. With so much of steps, it's going to even 10 products is an uphill task. But then there are certain reasons certain reasons where this Indian industry has developed. I think that becomes, so it's a kind of an ongoing process. Let us start somewhere, start with 1 product, 2 products, 3 products. I think one thing what initially when I say is when we start, knowing how to develop a product, maybe first or second we might have a problem, but then it becomes very easy. It also gives you an opportunity to develop innovative dosage forms. What is the customer's patient's requirement? Now, talking about theory and review of literature, the data provides, whatever the data, why this is important, review of literature. We need to knowledge to support our development process. How do you design? What is our design space? Then critical materials like API and critical excipients, what should be their specification? What should be their attributes? What should be our manufacturing process? How do we going to manufacture, whether it's a dry granulation, wet granulation, direct compression? Now with this kind of a literature knowledge, this becomes very easy. Manufacturing controls. Now we also look at from where we can get all those review of the literature. I think any MPharm students, I think for him it's a very easy, but then there is need to do it. We are looking at pharmacopoeias. Some of the reference books are there. US FDA approvals. Online computerized search, electronic database. Today it is so easy. When we were doing MPharm, I understand I don't know how many kilometers I had to go to like Ames library, then we used to go to our IIT libraries. Almost a month we were going only on scooter, half of the time commuting and searching where book this particular data is available. Journals and publications, FDA CDRs, Orange Guide, and lot more things. This is how we can, these are some of the references where you can do the literature survey. Today it is so easy, just Google and you can get many data, but then you also need to understand which data is relevant to you and which data is authentic. Just Google looking, as you say, when you go to a doctor for a certain disease, you will have already done the Google search and gone. But then still when you're not being okay, then you go to the doctor. This is something like this. Now, coming to formulation studies, why do we require a pre-formulation studies? Our development requires extensive chemical, physical, and characterization of whatever we do. Actually, whatever product what we make, we should have a thorough knowledge of it. What starting materials we are going to use it, how we are going to use it, what are the packaging material, what is the process, what is my finished product, what are the improvements I can do on a finished product. This is where you start with a pre-formulation studies by understanding, and you don't have to spend any money for this. You have to do a literature survey. What is the available data is already available. You need to understand what is the chemical, physical characterization of not only API excipients and packaging material to how to have a development process to start. This is what we're talking about, quality by design. And the whole process starts with pre-formulation studies. The purpose is to gather knowledge about all your API excipients, manufacturing process, finished formulation, demonstrate that the desired pharmaceutical performance has predictable therapeutic response. Whatever development you do should have a predictable therapeutic response and stability throughout the shelf life. The finished product has necessary, whatever we have designed, quality, efficacy, and safety, your finished product should be of that particular standards. Very important for academics who are present here. We do a lot of studies, and what we industry people feel is it cannot be applicable to a manufacturing site. And even in our own R&D laboratories, we feel that sometimes the process is so complex, it becomes virtually impossible to scale up to the industrial site. So how please understand, when you do the development, you should also need to, at the back of the mind, you should also need to understand how am I going to scale up to the commercial production. We need, as far as API, we need to identify and study properties of API which has an impact on formulation designs. So there are various parameters. I don't want to, anyway, I'll be giving you the PPT. There are various parameters. So evaluate and understand the specific excipients which can give you optimal performance of your finished product. Now, this excipient what you choose, it should be compatible physically, chemically, and microbiologically with the API, not only with the API, with the other excipients as well. Now, you know how inadequate stability can cause poor quality, safety, and efficacy should be stable throughout the shelf life. Loss of potency can lead to degradation products. Due to degradation, products can develop even during processes. And all this has to be investigated thoroughly. Instability, now you have seen many cases. I think one of the biggest brand in this country, this has to, not only in this country, in this whole world, and we have experience in Karnataka, the investment of 1,000 crores on the plant gone down the drain because the degradation products were found to be cancerous. And so much of, I think we had another issue of the talcum powder, I think baby powder, I think most of us know, I think how many billions of dollars were across the world was spent on settling all those claims. I think over a period of time in India also we'll see, I think it has already started with vaccines. So I'm sure these are the indirect costs also we need to understand. We also need to understand what are the external factors. We are looking at temperature, humidity, light and oxygens, which manufacturing process, packaging material, which has on the overall stability of the product. API and excipients, as I said, I won't go much into detail, study chemical, physical and microbiological stability. Chemical stability, very important. Physically, although microbiological stability is not that important as far as tablets, solid dosage forms are concerned, but then we need to understand and recognize proper limits. Talking about solid state stability, now when you talk about tablets, it's a very complex process. The problem what happens is, in a solid stage, unlike solution, the interaction between the molecules is very minimum, less number of molecules. So it is very, the reaction is very slow. So the kind of results what we get, it takes its own time. Then there are also multi-phase reactions are there, which are difficult to interpret and predict. Now, what we can do is, we can do it under accelerated conditions, whether how this combination of API and excipients and packaging materials will, what are the degradation mechanisms are there. We can study in higher temperature, but then again, there are limitations are there. Whether is there kind of a more than one degradation mechanisms are there, or whether it is following that Arrhenius kinetics. Temperature. Now, temperature again is very important, as I think sir had told about. We always say the storage condition was not good and all that. I think most of the products, if you see the labeling on the product, we are looking at, we manufacturers are very smart. We'll always store below 25 degrees centigrade, knowing fully well that there is no place in this country where storage at 24 hours, 365 days is possible less than 25 degrees centigrade. So, again, I think coming back to the development, I think that how we test this effect of temperature on the stability is by doing isothermal testing on the, and degradation at higher temperature. Now, one of the most commonly employed process. Now, it's again, as I said, it is based on the principle of Arrhenius kinetics, which states that the reaction kinetics is based on the temperature, but then it is not always the same thing. There are many times the Arrhenius plot is not linear, and this shelf life prediction based only on the Arrhenius kinetics can, as I think most of you can know, it can be used only if there is no significant amount of degradation products are produced. But then most of the time there are multiple mechanisms there, and that can be, if there is more than one peak in a HPLC impurity analysis, then we can be very sure that there is a change in degradation mechanism. So that's how you can study the effect of temperature. Then normally you can use multiple stress conditions to study the effect of temperature. Again, next coming to what is the other factor of, that's another water on the stability of API and excipients. So how do we study that? Now, the moisture or the water can come either from the API, which can be chemically bound or unbound. In same case with the excipients, it can get also introduced through a manufacturing process and the environmental conditions. Now, the effect of water. Now, what water does is, as I said, in the solid state, the interaction between the molecules is very less. It can accelerate that kind of reactions. And because of a smaller size, low molecular weight, low density, and higher dielectric constant, it makes that molecular mobility much more faster. So it can increase your degradation reactions. Now, in cases where the water can affect the stability of your drug product, and the classical example again is paracetamol. And the problem with that paracetamol is, when we compress, normally you should have 2% to 3% water content in your granules when you compress. Then what happens to that 2 to 3%? That gives rise to the degradation products like 4-aminophenol and para-aminochlorobenzene. Now, what is as I said, it can behave as a solvent, as a reactant in the hydrolysis reactions, increase rate of solid state reactions by acting as a reaction media. I'll tell you, there are certain excipients are there. I think one of the classical example is colloidal silicon dioxide, where this excipient preferentially binds to the water compared to the other materials, and that can sometimes be utilized. This kind of excipients can be utilized to increase the stability. Now, how can we evaluate the effect of water? By directly incorporating into API or in a mixture of API and excipient. And also you can, this is what we call as accelerated humidity conditions, where you can close system through controlled RH chambers. Now, how do we, once we understand what is the effect of water on your stability of API and excipients, based on that, we can design our process. Whether do I need to design a process which is low RH process? I can give example like sodium valproate, amoxiclav, enalapril, cefuroxime. Enalapril is a classical example where, although it is not hygroscopic, but then very sensitive moisture. You can we employ a moisture barrier coating? This happens with most of your fluoroquinolone antibiotics, or even azithromycin. Selection of excipients which bind to water. As I said, you can add some excipients like aerosol which can preferentially bind to water. So although the water is there in the formulation, but it is not available for your reaction. Can we employ a moisture barrier packaging, whether alu or a strip packing or a container with a desiccant? Another important example which most of us ignore is a mechanical strength. Most of these API and excipients are present in a thermodynamically stable crystalline form, but some of them also are there in the metastable amorphous form. Now, what happens with the stress is, during this process of milling, grinding, sieving, it can lead to formation of amorphous pockets, which can, as you know, the most of the amorphous state, the stability is little bit compromised. Crystal defects, increase in surface area and increase in contact between API and excipients. Now, all this rate of degradation reactions and reduction in dissolution rate, it's crucial to keep the crystalline structure as intact as possible. So this is mechanical stress. Understand the effect of mechanical stress and temperature, water, and do a risk-based analysis. Then we are looking at API, how do we screen the API excipient and do the compatibility. Now, when you talk about excipients, although we say it has chemically it is inert, but then they are rarely so. Now, why do we add excipients? Now, excipients has to be, when we add any excipients, we need to evaluate for safety, and is also included to whether to need to protect the API, support or enhance stability, or enhance safety and efficacy. Now, when you understand all these factors, you can critically select which excipient has to be used. Now, how do we select the excipients? Based on their functions, whether it's a diluent, disintegrant, binder, lubricant, glidant, coating agent, acidic alkalizing agent. Can we add acidic and alkalizing agent in a tablet? Anyone? Anyone? Yeah, one person. Yeah. Okay, sir, very important aspect. I think you mentioned about a lot of dissolution failures and assay failures. And one of the more important molecules are proton pump inhibitors, which fails in dissolution and thing. This is where you use that many of us don't understand. We can use this alkali and acidic ingredient excipients to enhance their stability and dissolutions. Another example is telmisartan. So these are some of the things. I think if you look at proton pump inhibitors, we are talking about they are unstable in acid media. So two things. When we take the tablet, we directly gets into the gastrointestinal tract. So we need to protect it from gastrointestinal tract. Before that, what we are doing is to protect that. We are using enteric coating agents that themselves are acidic. So how do you protect this API from the coating agent? This is where the assay failure and dissolution failure happens. So what we do is we add certain alkalizing agent within the core of the tablet, number one. Second thing is also do a seal coating so that you keep a barrier between your core and the enteric coat. I think that solves most of your problems. Compatibility with other excipients, API and impurities, and what are the degradation examination. Having known that, how do we select the excipients, we go towards what are the incompatibilities. Now incompatibilities can be caused by undesirable reactions with visible in form of physical, whether it's a color and appearance. One of again the classic example is you don't use, whenever you have a primary and secondary amines as your active ingredient, you don't use lactose. I think if I remember well, it's a Maillard reaction where it turns that brown color. Mechanical, as we have already seen, chemical increase in impurities, microbiological. So there are certain undesirable interactions which will be visible. Now, how do we prevent those kind of undesirable reactions? By selecting proper excipients. I talked about colloidal silica. Another change in microenvironment of pH. Sometimes MCC, which shows higher surface acidity and high moisture level. So again, when you look at some of the excipients, at minute amount also they can increase your degradation. So select the excipients, we need to filter it out. This is where I talked about every stage you understand what is selected, what is not selected. So over a longer period of time, you don't waste your time. It's very simple. When we started some kind of a development, okay, you got some formula from somewhere, we did that batch, put it for stability, and after 3 months or 6 months, we say it is a failure. So you lost those 6 months. When you talk about evaluation of API excipient, how do we evaluate what can be the kind of interactions? You can expose that to higher temperature and humidity, as most of us we do. Exposure to UV radiations and high intensity light. Exposure to peroxides. If you feel that API is susceptible to oxidation, then differential scanning colorimetry can be very important. Having done all the pre-formulation studies and how we can do the risk management. How do we understand each of those factors? What is risk? It's a combination of it's a pure theoretical definition. It's a combination of probability of occurrence of harm, and once the harm has occurred already, what is the severity of the harm? So based on that, we can classify what is the severity. Now risk management is a useful tool. It's a scientific tool to in a development process, and we can make great product dossiers. I think today no product dossier is complete without a risk management and risk involved. You need to understand the risk assessment by stating the problem and understand how it can compromise the stability of the product. We need to understand that. That's by identification and classification of hazards, analysis and evaluation of risks. Now there are risks are of 3 different types. When you look at, if it's a high risk, if you find that, say for example, I talked about a primary amine and we are adding into with a lactose, it becomes a high risk because you know by physical, the brown coloration is bound to happen. So that we say high risk, so you avoid that, so you understand that risk and reduce the risk. It's a medium risk, it may be needed to reduce the risk. Risk is sometimes acceptable, but investigation might be required. If the risk is low, no further investigation is required. Now, next slide, I'll show you how do we assess the risk. Now, when you say about risk assessment, we are connecting the attributes of all your materials, API, excipients, with your processes to the finished product attributes to correlate and evaluate the potential impact each parameter will have. Now, as I said, risk has to be developed at every early stage and updated during, as we understand the knowledge, we get the knowledge, this risk assessment has to be, sometimes initially we start with the say, this has a particular high risk, but later on as we understand the process, the risk can be, say, okay, it's a medium or a low risk. It is also prudent that finished product CQA selection of adequate manufacturing process, development process, so that risk assessment it could be defined. Now, critical CQAs identified for tablets to have potential to be impacted by process and formulation process variables are assay, degradation products, impurity level. I just list it out, I'll not go through directly. And each, now quality attributes, now these are the 5 which are very critical, but then there are certain non-critical attributes are there which are not considered as critical, but then needs to be evaluated for their standards and reference product statistics. Again, I've listed out some of the processes and steps where the risk assessment during the manufacturing process is required: the sieving, milling, drying, compression, coating. Now, coming to the API, we need to understand what are their chemistry, therapeutic profile, pharmacology, reaction degradation mechanism. There are various degradation mechanisms are there. Then what are the critical attributes which has to be selected and evaluated. Now when we already defined the finished product specifications or the attributes, now how do we relate that to the API attributes? I think that needs to be evaluated. Now example of, as I said, I think this is a very important tabular column where you can do an assessment of typical properties of APIs where needs a, whether how can we classify whether it's a major risk or whether the risk is there or not risk is there. Now if you're looking at this, take example of solubility. Now considering when you talk about solubility, assay or dissolution, when the drug is already soluble, then that attribute is not so important. If it is insoluble, then it is important. So similarly, we have for particle size distribution, hygroscopicity, water content, chemical stability, flowability, compressibility, polymorphism, apparent density. Very important. I think that's where I think we in India we need to develop this particular culture where innovator or the products, let's not say innovator, but the leading products in the market, or the leading company, when we develop the product, we need to understand their all attributes, what are the properties. I think I listed out some of the properties where we need to understand and make a report and make it in writing. Now, why do we require this kind of data? This is information is used to guide development process and set the finished product specifications. Need to purchase at least three different lots of higher and smaller batch sizes to understand each strength and for evaluation. Important. I don't know how many of us go through this patent search when we develop the product. Very important. Why it is done? The patent is granted for protection of intellectual property. I am a scientist, and whatever development I have done or whatever new I have done, if somebody copies and takes his benefit, will I be happy? Absolutely not. So that is where the patents are granted. And it gives, I think that last line on this slide give you why this patent landscape is important. It's generally given for 20 years for some, whether it's for the whole world or a particular country or the territory. It can be issued for API, finished product, manufacturing process. It's a valuable source of information to avoid infringement. Last line, gain insight into previous work which is help to guide alternative novel approach. Then, having done so much of studies, we need to understand, with so much of data we have for API and excipients, from where do I buy this API? How do I have a proper source? This is where you can do this kind of steps. Every manufacturer will have his own some amount of small kind of a data or catalog. And that catalog, request samples, specifications, method of analysis, certificate of analysis. We should, as an entrepreneur, I would say, always for every material you should have 2 or 3 manufacturers to reduce your risk of supply chain instability. Very important slide, especially even the existing manufacturer if you are looking at doing a vendor validation. Whether this particular manufacturer has DMF availability, registration or drug manufacturing license, especially today, for as Dr. Ishwar Reddy ji has said about excipients. Today in our country, I would say half of the excipient which is used by the industry is not licensed. I can make that standard, whether it's a, when I say not licensed, is a drug license. Many of the major products are licensed only under FSSAI, not under drug manufacturing license. And I told on multiple forums, we should encourage all these excipient manufacturers to comply with the GMP and also start taking manufacturing license, either from the regulatory side we should, or from the companies who are buying these materials. Otherwise, you say we don't buy from you. Compliance with the pharmacopoeia standards or the manufacturers, our own specifications which are approved by the regulatory authorities. What is his impurity profile and stability, potential polymorphic form, commitment to physical specifications, statement of non-patent infringement, important, whether he has GMP or GLP certifications, compliance to EHS requirements, extremely important. When you are doing a business and tomorrow for some, how many times we have seen that our critical API manufacturer has got into pollution control issues and his factory is closed. Now, because his factory is closed, can I close my business? Absolutely not. I think this we can do at a kind of a paper audit, and this is where kahin se to shuru karo bhaiya, vendor validation ka audit ka kahin to shuru karo. You buy those, once you have evaluated the API manufacturer, you buy the API. Second approach, never mix batch numbers of API in PQ and pilot batches. Once you purchase the API, do the testing as far as physical parameters are concerned and chemical parameters are concerned. Whether these are complying with your requirements. Then the container and the closure system. What is the kind of packaging? Having known the pre-formulation, what is the kind of packaging, what is the material composition, type of resin which is being used, what are the materials and suppliers, liners and seals used, cotton and desiccants. Again, coming soon, even for primary packaging material, drug license, registrations, DMFs, coming soon in India as well. Important, I think this is where we have one of the major issues where letter of access for the regulatory authorities to view DMF and registration dossiers. Having known now with this, we know what is AI, what is the excipients, what is the kind of packaging material we will be having. What we need to understand is next the process. When you're talking about with that knowledge, what is the kind of suitable manufacturing process, whether we have a wet granulation by aqueous or non-aqueous or a dry granulation. What are the parameters we need to understand in wet granulation, whether it's a high shear or low shear mixing, FBD spray procedure, determination of order of mixing, determination of fluid addition, determination of granulation time, torque endpoint, drying parameters, LOD. All these are very critical factors. We need to understand that every process, all this can be done only we know what is actually what we want and what is the kind of raw materials what we are using, what are the properties of the raw materials. Looking at manufacturing process, at every stage of manufacturing process, what are the physical properties of whether granulates, compressed tablets, coating, we need to define everything and how do we going to achieve it. Finally, with all these factors, we are going to make a master formula. This is what we called as a master formula, and do at least 3 months initially a stability, accelerated stability, where we understand what is going to be my results. Important analytical method evaluation, whether it's a dissolution testing versus the innovative product, uniformity of dosage units, analytical method validation of assay dissolution content uniformity. Having done that, initially whatever we have done, next is, see, our aim of a development process is to make the product, a finished product from at every stage, every tablet which is coming out of your factory should have a same quality, robust process. Finally, a secret what every entrepreneur always look at is how do we reduce the costing. When your output, when your process is robust, your manufacturing process is robust, your output is very high. This is how you reduce the cost of your production of your product. This is what government wants us to understand. When you look at process optimization, at granulation step, what are the various parameters are there, drying step, optimize all this kind of parameters, then make a report called as a process optimization report. Sorry, I'm running too late into my thing, but so I'm just skipping some of the things which I think most of you already know. Finally, we are looking at how do we establish in vitro, in vivo correlation. Extremely important. Otherwise, this also we need to understand how we can establish an in vitro, in vivo correlation before we go for even bioequivalence studies. As sir had talked about bioequivalence study, which is costing around 10 to 12 lakh rupee. And once you send your sample for bioequivalence and if it fails, once you send the sample for your bioequivalence study, you have to be 100% sure it will pass by way of your knowledge about your product, knowledge about your process. Perform in vivo in vitro bioavailability study where relevant. Establish a level A and C correlation without adjusting dissolution parameters. Initially you do that. In case you fail, then you can always try to adjust the dissolution parameters depending on what is the kind of API what you are using. Adjust only if necessary. Then we are looking at scale-up batch. Scale-up batch is prepared if you find that the larger batch is going to have some kind of a problem. So to understand what are the kind of problems will come and what are the kind of risk mitigation mechanisms we are going to employ. We can, I think it's a kind of a subject of debate whether the PQ batch and scale-up batch may be evaluated as a single batch. And scale-up reports also forms a part of the overall development report. Process qualification, I will not go much into detail. Prashant is going to take up, but the only thing what I can say is process qualification batch is manufactured in order, see if any problem arises, so in the production stage, so you can suggest a solution prior to making a pilot demonstration or exit batch. Now normally the scale up to the pilot batch size or the 70% of the batch may be combined with qualifying the manufacturing process. And at this stage, full manufacturing documentation is prepared along standard procedures. Now these are some of the, what are the kind of documents, what are the kind of facilities we should do at the time of process qualification. And finally, I think with various steps of final review, protocols, key steps, operating conditions. So you prepare a process qualification report, and that again also forms a product development report. And there are various who are the people who should be involved. I think some of the key people should be involved with, not only from regulatory QA but also the production staff. Then you make similarly a pilot batch what are the kind of production facilities, what should be the batch size, what should be the batch document. So these are the various, I don't want to get more into this. Then the bioequivalence, we get into a bioequivalence study, and we need to, as sir said about, we should have minimum, as far as the Indian conditions are there, minimum 16 patients are required, and whether we need to understand whether we require fasted and the food effects study also may be required. And normally, the bioequivalence study is performed on the highest strength, and then you also do with that for multiple strength product, you do a in vitro testing is conducted at 3 different pH media of the lowest dosage forms, and also do a similarity test on the dissolution results. Extremely important. I think this probably I took it recently from one of the research article. And extremely important, before even you submit your data, development audit is extremely important, especially when you're looking at a larger company. The CQA, the corporate QA, should also look at the development audit. You should audit all the raw data supporting the development report, audit plant and laboratory documentation. You should go to every place and audit what is happening. I think this is what even the CDSU has been doing recently. When you file a application for your bioequivalence as well as the new drug application, they go and audit the facility where the development work has been done. And recently there have been many instances where the new product launch itself has been stopped because of the audit issues. Review of SOP system and update the level, review CGMP of manufacturing process, evaluate and develop a in vitro in vivo correlation, level A where possible, and complete product process validation protocol completely and signed. This is extremely important development audit. Then we go for a validation batch just before commercials, where we can do a validation for 3 consecutive batches and prepare a report, and it should be 3 consecutive commercial lots. This has also become the requirement today in India. Prepare a slow process validation report, extremely important. Last 2, we should show intra-batch similarity and inter-batch similarity with respect to bio-batch and commercially validation lot. Extremely important. Require a revalidation report. What are the major issues where your process has to be revalidated? So formula change is there, or a process change is there, or the equipment change is there. You revalidate with the new formula, process, or the new equipment. And if it is a minor change, then you can follow the scale up and post approval changes rules, level 1, 2, or 3. I think with this, probably I'm doing injustice to the topic which is given to me. I think this requires a 2-day workshop. This topic alone, 2 days workshop. I have tried my best to put that into a 45-minute or a 1-hour program. Thank you very much for patient hearing. Only thing what I can say is, make mistakes in the small level and make profit at the larger level.
Thank you very much, Mr. Harish Jain ji. That was almost like a master class, very elaborately done, and we did really enjoy it. 21-step PBD. It was a vision given by Dr. Ishwar Reddy that quality control should be vanished from pharma industry, because we are designing the technology and the quality into the product. So I request Dr. Ishwar Reddy to come over and hand over a bouquet and a certificate to the speaker of the day, Mr. Harish Jain ji. Thank you very much. much. Actually, for Mr. Harish Jain, there are some favorite names, that is his family members. After that, it is paracetamol. He had to touch on paracetamol point. Well, you don't seem hungry actually. All of you look so energetic. It looks like we can continue with the... But anyway, we are very careful about our health and diet. So we'll have a lunch break exactly for 21 minutes. So you have to please go right to the back doors and then go to the 4th floor. Okay? So please come back after 21 minutes. Have a nice lunch. Thank you.
ಒಂದು ಲೇಪಲ್ ಬೇಕು ಮ್ಯೂಸಿಕ್ ಕಟ್ಬೇಕು ಲೇಪಲ್ ಬೇಕು ಒಂದು ಲೇಪಲ್ ಮೈಕ್ ಬೇಕು ಒಂದು ಬೇಕು ಲೇಪಲ್ ಬೇಕು ಚೆಕ್ 1 2 ಹೇ ಚೆಕ್ 1 2 ಚೆಕ್ 1 2 2 2
All. Good afternoon all. Please start settling down. We'll start in about 3 to 5 minutes. So this time I am giving a range, 3 to 5 minutes. Yes, good networking. I didn't shout out in the dining area for finishing the lunch and coming fast to auditorium. Any reasons you can guess why I didn't say that? Normally we go around in the dining area and say, please finish fast, come to the lecture hall. But this time I didn't say it. Because I was confident, after a sumptuous lunch, you are waiting for the AC hall to come and relax in the chair. So I never even said it. Yeah, all of you are almost here. That's very nice. Please flash messages on the WhatsApp. We have to start. So we had one masterclass-like session by Mr. Harish Jain. Now get ready for one more very interesting session. I request Mr. Prashant to come on. Please give a big hand for Prashant. So it's a very, very great pleasure for me to introduce Prashant. Prashant is my Chetty Dost. By Chetty Dost, I mean we were primary school friends to that level. And later on, we became batchmates in M.Pharm also. So Prashant, I am very happy to see your progress. You are now one of the, what do you call, the best QC QA guys in the country. This I came to know from Mr. Cheru Ramesh. Cheru Ramesh is, I think that company name is Alpha Tech. So the owner of Alpha Tech or the prime mover there is Mr. Cheru Ramesh. He is my classmate. So while chit-chatting, he was referring to you spontaneously and explaining about how good you are in the various QBD aspects and also in quality assurance and looking and checking into various QC aspects. So throughout his 33 years of experience, Prashant has contributed significantly to companies like Cipla, AstraZeneca, Orchid, Neopharma in Abu Dhabi, Chemwell, Microlabs, Strides, and his specializations are in QC, analytical development, quality assurance, and corporate quality assurance. So that's a new term, corporate quality assurance, at least for me. And his extensive expertise and leadership continues to drive excellence at Granules India Limited. Please give a big hand to Mr. Prashant. Prashant, this stage is yours.
It's connected, no? Yeah, good afternoon, everybody. It's been a Saturday, that too a sumptuous lump which Sunil Chiplankar told, and the gathering is good. Many of the programs we see, post-lunch it will reduce by at least 50%. So at least the program organized by IDMA as well as CDSU and the KDPMA, at least the crowd I see is remaining the same even after lunch actually. That's a good sign. Sunil, he tried to tell many things in the morning. He made all of you very active actually. Not that you people are not active. He made definitely all of you very active and he continues to do that. First of all, I would like to thank the CDSU team, the KDPMA team as well as IDMA for giving me an opportunity to present a topic on process validation. Majority of the things of the stages of process validation as defined from the FDA regulations and also the Schedule M related guidance and also the different guidance from various regulatory agencies, the definitions also we'll touch upon. Before that, Dr. Ishwar Reddy has given a very good insight of the various, his viewpoints on many of the things of the industry. And industry always, we talk about continuous improvement, I'll touch upon. That is what all process validation, pharmaceutical quality systems is designed for. Harish also touched upon some part of the pre-process validation as my terminologies. So a lot of activities which have to be done at the design space, including the QBD, including the DOE, has been mentioned. So I'll just take a couple of minutes to tell one video of Mr. Rajiv Bajaj. So I was just going through maybe about 5-6 months back on a small video. He meets one of his mentor called Imamichi-san. So in Japan and Korea and that region, basically, everybody, elders are called as, like we say, sir, they are called as sans. So when he meets Imamichi-san for the first time, Imamichi-san asks him, what do you do? So he says, I am the managing director of the company. You are a managing director, that's fine. But what do you do? What do you do in the company? Another question comes from him. So he gets confused. I am a managing director. He thinks to his mind, what do I answer? He says, no, I manage the entire profitability, PAT, whatever, sales and other things. No, but what really you do in the organization? There are people for that. What you do? So he gets little puzzled. Okay, then one day he invites him to his factory. So in the opening meeting or the introduction, so Imamichi-san is there, Mr. Rajiv Bajaj is there, the head of the site is there, then head of marketing is there, the entire core team of Rajiv Bajaj is sitting in the meeting. So he asks every person. So when he talks to the operations head, what are you? I am the operations head. What do you do? So he says, I am president for operations. You are talking about your designation. What do you do in the company? So that again a question starts. So he interacts with all the senior team. He makes them feel that they do nothing in the organization. Okay. So everybody talks about their designation. Everybody talks about their role. But he also talks to the R&D. He does the product. That is the motorcycle or whatever products they do. Do you touch the product? I don't touch the product. You are not touching the product. The operations said you are not in the shop floor. So how do you ensure that things are right? So they give their own explanations. Finally, he says, all of you are paid for continuous improvement. You are not paid for your designation. You are not paid for the activities, but you are paid for continuous improvement on a daily basis. So having said that, the Drugs and Cosmetics Act implemented way back few decades back, slowly the Schedule M also came into the place. The revised Schedule M also has come. So all of us in the industry, be it from the various companies, be it from various functions, we all read. The challenges of implementation is not that easy as we speak. So I just have a favorite word of mine. It is easy to say the world should be full of peace. So if somebody gets a Nobel Peace Award, it's fine. But it's easy to say. But a population of almost 7.8 or almost 8 billion, to make it peaceful, imagine the kind of percolation which should happen. Imagine the kind of diversified people, diversified country to bring a common goal of peace. Just let us think to ourselves, is it easy? It's not that easy. So though we see a lot of guidance either from the FDA or from the European agencies or from Brazil and the various global regulatory bodies including CDSCO, the guidance, I appreciate Dr. Ishwar Reddy's statement. One word of the product and process knowledge is extremely important. So today, people do extremely well in the interviews in organization. I'm talking from a manufacturing organization perspective. People do extremely well in the interviews. People do get a job. Okay? So the interview questions are completely different. But a product and process knowledge and experience is again completely different. So the penetration of the knowledge into each individual in the organization is an Herculean task. Though we say that we build facilities, we build world-class facilities, we spend millions of dollars on import equipment, okay, in terms of the HVAC design, the best of the water system in the world. Finally, it is people who matter in the pharmaceutical industry is my belief. You can have all things in place, without the right set of people into the organization, it becomes extremely challenging. And again, it's people, again, it's not a head count. So we can say that we have 10,000 people in our company, we have 5,000 people in the company, we have 2,000 people in the company. The most requirement is the passionate people who has a common goal is also extremely important. So when we put these filters in a mindset again before I start, so you narrow down that list of people. So though we say yes, number of people, as we put filter, okay, in terms of improvements in product and knowledge, you will finally see in any organization, there are very, very few hand set of people which can be countable, which could be running into double digits or slightly higher 3 digits, who are really passionate driving a cause in the organization. So having said that, process validation is the third part of today's discussion. Of course, the last part is on the APQR. So I'll just take a couple of minutes on to tell the content of this session. We'll talk about the importance of process validation, then the differences between the validations and the qualifications, the entire life cycle approach, the statistical analysis which are extremely important, and what are the citations either from drug control or from the FDA for various organizations, and some guidelines references. So I'll just stop here for a minute. Just I'll keep changing. I'll not speak extremely in the same flow. If you see many of the FDA 483 observations today, which is published by the FDA in the year 2023, September, when you do a Pareto analysis of these observations, so the biggest challenge for the industry is on the lack of adequacy in investigation skills. If you summarize our own terminology to that, okay, it's basically about the investigations not appropriately done by the organizations. So what are these investigations? These investigations could be either the manufacturing investigations or the analytical investigations. So if you again do a very high level root cause, not the technical root cause for this issue, at an organizational root cause as we call, the product design comes as a most challenging root cause when you do a YY analysis. The moment the product development, the design and development which Dr. Ishwar Reddy was talking about, if it is not done in the right perspective as it is supposed to be done, he also mentioned, I remember that the duration of issuing or granting a state license, that's the language he used, people expect to be done in days to the regulators. But the quantum of data required right from the design stage to the stability data as a part of checklist of the drug control department is huge. The time required right from designing a product, right from the QPP as we call, to the availability of the third month stability data, the time frame itself is huge. So there are different thinking on these investigational failures. One is the timelines required for development, it is cut up to the stability. This is one of the biggest contributors. The other one is the knowledge on the product itself at that stage of the design and development. Pharmaceutical operations are like a relay race. It cannot be by a single person who can just take the baton and run 100 meters or a 400 meters and say he has won. Pharmaceutical operations are the entire value chain contributes, right from, in this case we talk about the topic for the day, the scientists who are involved in the R&D part, the analytical people who are involved in the analytical development part, the validation team in the R&D who does the analytical validations, the document preparators, the specification designers. So it starts from everybody. So once this R&D activity is done, there is also a tech transfer group. This entire knowledge cannot be within the one group. by the time the product comes into commercialization, there are multiple departments involved by the segregation of the functions and the roles and responsibilities, as we call in the organization. The knowledge within the R&D also has to be transferred to the functions, that is to the manufacturing operation setup. For that, you need an intermediate team called the Technology Transfer Department. So there are many videos you can see on the WhatsApp, some management programs where we have a set of 25 or 30 people standing in the queue. There is one person, the trainer, he tells something to one person. Other people do not know what he has spoken. So he has to do some action, and then the action had to percolate to the end. So the person does some action on the first person. He demonstrates to the second person, others are not seeing. The second person does his own fashion. He understands something, he does in his own fashion. Then he repeats to the third person. The third person understands completely different. So when you see this entire chain of the first person who has demonstrated something of a dance or whatever body language you call, to the end person, it is completely distorted. It's not the same. How many of you seen those kind of videos? Multiple videos are there, especially on the management front. So what does this mean? So whether what we talk, verbalize, the messaging happens in the line will be completely different. This is one way of inference. Second, if you apply that video, all those kind of videos into this concept, as long as the knowledge transfer has to happen till the commercialization, there is always filtration happening. Though you say the R&D team has a complete knowledge on the process, the business doesn't end there. It has to come to the entire vertical of different sites. Suppose you say the same product is done in different sites, for example. I'm just giving an example. So the knowledge transfer is not in one place, it's across the value chain. So imagine the challenges of the pharmaceutical industry, how the information should not be diverted, the information should not be distorted, how the documentation knowledge, the knowledge management as we call, the knowledge management has to be gathered. It's a Herculean task. Having said that, there is always a variability of people. So though we say process validation, I'll come to the definition in other parts. The people change in organizations. People at different levels change in organizations. So you are adding another variability into the entire discussions what we are doing today, that the knowledge is in pockets, knowledge is not converted into document, so execution becomes an extremely challenging situation. Why I am telling all these things is about the biggest concern on the investigational observations which as pharmaceutical manufacturers we get. So having said that, I'll just take, Manjunath is my good friend, he's here. Goni just now told, you have to make something interesting, you have to make some comedy in the session is what I heard. Okay, I'm bringing the name, sorry. So I'm not a comedian, first of all. So let me give some examples what I can think as we speak. A new daughter-in-law is into the house. She cannot learn cooking. Though she observes mother-in-law doing or a grandmother doing, whatever it is. Ladies, not me, I'll just give a silly example. Just by seeing a person, a grandmother or a mother-in-law doing a cooking, can the daughter-in-law learn from the next day? Is it possible? Same thing, there are multiple master chefs, if you see on the YouTube, multiple fantastic people, great people, Michelin stars. We cannot say, I just watched their video and become a master cook tomorrow. Correct? So the same application if you do here, it's not that easy, it's a task. That's why I use the word passionate people are required. People who are having an ability to understand, ability to contribute, ability to learn continuously is a factor what we are talking about. Having said this background, I just tell some of the definitions here. So why I use the word, if you see here, the process validation definition as given by the various guidance. So I'll not touch upon everything. The key elements, if you see, the process validation is a systematic approach ensuring that a manufacturing process consistently produces a product meeting predetermined quality attributes. So the word consistency is extremely important. So the variabilities have to be reduced. the variability in the equipment, variability in the materials. Morning we spoke on the, I think Hari spoke, touched upon vendor qualification. It came multiple times in the earlier two sessions. So the variability in the materials, variability in the people, variability in the practices. So everything contributes to this consistency is what I wanted to tell. So there are different definitions from the various regulatory bodies, more or less, not more or less, it's, the meaning is essences of all these definitions are same. In the interest of time, I will not touch upon that. So if you see the objectives of process validation, one is to confirm the consistency. That means we produce 100 batches in a year. All the 100 batches have to be consistent in the quality. I would like to tell you, when I joined this industry about 33 years back, I always, like how Mr. Mudda spoke about Dr. Iyer, we all are SIPLites, many people are here, we give our tribute to Dr. Bami. When I joined, there were a lot of visitors used to come, which I can recollect now. He says, in Cipla, we are not interested. I'm bringing some company's name. It's from the heart, actually, not to talk something different. In Cipla, we are not interested in this purity or assay of 99.5%. This was way back, 33 years back. ICH guidance were not into existence during those times. We are interested in what that 0.5% impurity is. So the ICH guidance came way back of the Q7 came somewhere in 1999 and early 2001 to be precise. So the industry had geared up the vision. Some of the visionary leaders in the industry were thinking on those lines much before regulations were in place. So if you really see the consistency, how the consistency is managed, the mitigating the risk, once the product is into the market, once hundreds of batches are in the market of the same product, if at all the design and the development, as told in the earlier meetings, if the stability issue happens, there will be a high level of market recalls. The cost of market recalls will be significant. It will impact the PAT significantly. It impacts the reputation of the organization very significantly. So mitigating the risks, you need a very robust process validation methodology and the system to be in place. And having a control, basically you need to have the control on the manufacturing operations to ensure the consistent quality is there. It also enhances the product quality. So how does it enhance product quality? It's a complete cycle chain actually, or in the entire chain. So once the process validation is done for 3 batches, the terminology now is continuous process verification, which I'll talk about. So continuous process verification, again, it's on all the batches which you manufacture to demonstrate the process is robust and is in the state of control. So then comes the APQR. After reviewing the data, again there will be recommendations for some improvements to further enhance the product quality. So this chain, what we call, continuously improves the quality of the products and the processes, and of course the documentation towards that. So this also helps in the continuous improvement of product. And also last one and the most important is on the patient safety. So Mr. Mudda also told that a few decades back, we were all ensuring that the product which goes into the market is safe. So that is what is more important or the most important in the entire value chain what we are talking about. So the importance of validation, if you see, it reduces the variables in the manufacturing processes. We also use qualified equipments so that the equipment variabilities are minimized. I will not say reduced, it will be minimized. That's why we have the critical process parameters which are well defined in the batch records. To ensure the data reliability, basically, the calibration of all the measuring devices across the equipment and the equipment train is extremely important. If some malfunctioning of these calibration of gauges are there, it can result in a batch failure. It can result in the yield losses as well as batch failures. The other one is the validate process to define and verify the acceptance range that produce a product that will meet the specification. I'll just skip here. So there are several definitions where we loosely use the word qualification and validation. So these are picked up from various guidances and various literature actually. So validation is for the entire process which gives a high degree of assurance. The process or the facility will consistently produce a product meeting the specifications. So at the same time, the qualification part, if you see, it also talks about a similar thing which is on high level of assurance. It also talks about ability to achieve. It also talks about the equipments, the instruments, and the infrastructure and the utility qualifications as such. So we touched upon from morning different stages. This is a brief of basically what we spoke from morning till now. The first part, if you see here, is on the stage 1, which is called a process design. So I think Dr. Ishwar Reddy touched upon the majority part of this. The second stage is on the process qualification, where some part between stage 1 and stage 2, Harish also touched upon. And the third part is on the continuous process verification. So what are these terminologies if you really see? In the stage 1, if you see, the understanding of material attributes, what we call CMS, is extremely important. Each raw material, okay, how does it behave? What are those key aspects? What are their particle size differences? What are their, the bulk densities or the sieve analysis? All those things, material attributes come into discussion before even a product is developed. Compatibility, incompatibility, what work of excipients along with the APIs. So that also has at this stage of the design of the product. And also the design space in terms of the developmental aspects has to be considered there. The control strategies which are in terms of specifications, the test to be incorporated in the specification, how does the in-process controls happens, how does the specification controls for material in plus finished products are designed happens at this stage of the process. Other part of process qualification, where we see that the equipments which are used have to be completely qualified. The calibration of all these equipments, which we call the prerequisites before the validation, we need to have the calibrations in place, 100% ensured with some kind of a checklist in the documentation. And also the process performance qualification for the first 3 validation batches, and ability to apply statistical tools to understand the variabilities. So just about 30, 40 years back, the quality person used to be the king is what we understand. I have seen some people telling, I touched the material, I know this will pass or fail. It doesn't happen today. Again, I'm just telling what I heard about before I joined the industry actually. I touched the material, I know. I was in a company, my first company, I was traveling in a bus, okay. There was a solvent vendor, okay, who buys a scrap solvents from that company. One day he was sitting with me, he was coming in a bus. So I, being some inquisitive, asked, do you have GC in your, so GC is something big at that time, do you have GC in your lab? He says no. Then you are buying so many solvents, what do you do? I might put my finger, I know the purity. So this is a live example, 33 years back this fellow told. After a couple of years, he used to come regularly. Then one day I saw him in a Benz car, okay, coming in Bangalore, he became quite rich. So what I want to tell in this, the experience in those days by putting a finger into the solvent and check the purity. So GCs were not well there during those days. So these were the things of expertise. Today we can't rely upon that kind of expertise. Similarly, I see some companies, some people, of course, experienced operators. Sir, I touch the granules, I can say whether it can be cut or not. And rightly so. During those days, rightly so. It worked. No hard feelings or bad comments on such people. That spoke about their experience and pride. But over a period of time, the evolution happened, guidance, documentation, assurance, checks, all came from a regulatory world. So in terms of statistical tools, why I told these examples, just by touching the powder or by putting the finger into the solvent, you will not statistical approach. So today, when we talk about last couple of years or decades, there are different softwares which are available, including Minitab, where we need to see the variability within the batches, variability across the batch, variability within the validation batches, variability within the commercial batches. That becomes extremely important. It is more technical, it is more scientific, more rational. So if you see the guidance also, we talk about in the entire thing, it's all about scientific approach basically. The technical approach and scientific approach is very much required. So the third part is on the continuous process verification, which is never ending, which is as we produce more number of batches, your continued process verification has to be demonstrated that the product and the process is in a state of control. So here if you see, a lot of data collection happens. The sampling and the statistical tools are used for statistical analysis. There are certain tools like CPK, PK, and all those statistical tools have to be arrived at for each of every parameter, what we call the CPPs as well as the CQS. Then improvement initiative through the process experience. So always there are certain learnings which have to be incorporated so that the product becomes, the process becomes over a period of time. So we touched upon a development report in the earlier sessions. So the development report, if you see, which comes under stage 1, is the design stage. The development report should consist, it runs at least 250, 300 pages, even as a summary. Okay. So how the product is developed, what are the compatibilities of the excipients with, what are the challenges faced, what are the key process parameters which drives the product, what are the key critical quality attributes which have to be demonstrated. We touched upon a lot of things on what you call dissolution, for example. I think Dr. Ishwar Reddy told industry NSQ of 5%, lot of dissolution challenges. Harish also told about the acidic and alkaline part. Though we say a robust development has been done, though we say that continuous process verification is done, if you look at some excipients like magnesium stearate, for example, a magnesium stearate from one vendor, okay, vendor one I'll tell in the interest of this one, for a particular product, let us say metformin tablets, the dissolution need not be, even after all the validations, it need not behave the same way it is behaving for another vendor. So there is another variability. Now whether it is a specific surface area in the magnesium stearate, what should be the control, whether the limit is 4 to 10 or 4 to 8 or 6 to 8, how do we narrow down? A lot of challenges are there. In spite of these, controls in place. Next is the risk assessment report. Harish rightly touched upon that. So what are the various risks, how the risks are controlled, how the risks have to be mitigated. It should be part of this process design itself. The risk, he also mentioned about the severity, the detectability, and the probability, what we call on a matrix of 5 by 5 by 5. That has to be put in place. The control strategy of the material controls, the in-process controls, the product controls in terms of CQS, the control strategies have to be put in place. The master formula record which Harish was talking about, the contents of a final master formula which gets handed over to the operations team through the tech transfer vertical is a document which is extremely important. So the key elements for process design, again it's a summary of that. Developing the quality by design approach, basically where several experiments have to be done. Statistical tools have to be used at a QBD stage as well as the DOE stage at that stage. Then identification of the critical quality attributes and execute the product or process development. And finalization of critical process parameters is extremely important. Today, where manpower operators are involved, we say in the batch document the critical process parameters have to be kept in bold. The human error is the biggest challenge in the industry. To avoid the human errors in terms of not missing out on the critical process parameters, that will be in the bold so that there is no mistake happening at that point of time. The product or the process design space has to be controlled, and finally a control strategy has to be in place in the design aspects. So I'll just stop over here. Like we spoke about some products, even like ranitidine, for example, I think Harish touched upon. A company, I don't want to name, set up a plant here in Bangalore, in and around Bangalore. It was for a capacity of 10 billion tablets for a product called ranitidine. Today, if you see, this molecule does not exist. So the situation, in spite of the design space, what we talk about in terms of the 3 stages of qualification, all these were followed, for example. So the market dynamics and the technical dynamics is another aspect of discussion, wherein a molecule can get killed in the industry, irrespective of all these things in place because of the nitrosamine issue. So that facility, which was exclusively designed for ranitidine, was sold to some other company. So I don't want to get into those details. I just want to tell, in spite of this, in a pharmaceutical industries, the market dynamics, the technical dynamics changes. Just to touch upon another subject on nitrosamines, the guidance when it changes, like FDA came out with a very strong guidance on nitrosamines. So when you say the implementation of these nitrosamines, so though all these things were done for the products which we talk about, either a metformin or a ranitidine, so there were hundreds of batches which were recalled for metformin, both IR and ER formulations. So why I am touching this example is on the critical material attributes. These nitrosamines are formed especially on the NDMA which we speak. The limit is about 96 nanograms per day. The spec comes for a metformin at about 0.032 ppb levels. For example, the amino group interacts with a nitroso group in the formation of, with the presence of acid to form this NDMA. Levels we are talking about 96 nanograms per day. From a tablet perspective, we are talking at 0.032 ppm levels, for example. This particular interaction, though we have CMAs in place, the attributes of the entire specification of the raw materials which we talk, everything is controlled. Now, there are certain nitrites in one of the excipients which are not in the specification. So these nitrites at a level of say 20 ppb interacts with the amine of metformin and creates this NDMA impurity. So if you really see hundreds, if you see the literature, you will come to know, or if you Google, you will come to know hundreds of batches of metformin were recalled from the market. There was a lot of litigation still going on. So having said about NDMA, the regulators did not stop there. European agency came out with 14 different nitrosamines. It's not one, it came out with 14 different nitrosamines. NDMA is one we are talking about. Similarly, FDA came out with at least 7 nitrosamines in their guidance. So we have Mahesh, who is an expert in nitrosamines, also here. So the industry has to gear it up for all these aspects through a regulatory change process also. So that's the point I was trying to tell here. So we also have the QBD. Now Mr. Mudda also touched upon the ICH Q8, and today's discussion is all about a mix of ICH Q8, Q9 as well as Q10, which the revised schedule M has brought some elements of that, including the quality culture part. So if the QBD approach, if you really see the diagram over here, one is on the process understanding, next is at the design, then the scientific knowledge. This is most important. Scientific knowledge not in mind, scientific knowledge on paper, scientific knowledge transfer, so that entire process has to be done. And finally, a control on the process. What are the variabilities? How to minimize the variabilities should be part of this design document. So the steps of process design, we spoke about the QBD comprehensive evaluation, then the CQAs, the risk assessment document. So I'll just skip in the interest of time. So if you see the design, what you call stage 2, which we talk about, in stage 2, there are 2 different parts. One is on the equipment qualification also. So this talks about the facility as well as equipments, wherein all the utilities and equipment used for the purpose of the PPQ batches should be completely qualified. It should be validated through the appropriate protocols designed for both the utilities and the equipment. Then the plan also should contain what are the tests to be done. So what are the outcomes, the timing of qualifications. So what is the responsibilities of each function in the execution of the PPQ batches, and also which are the procedures which are applicable. The training of all these procedures to the people has to be ensured before the start of the validation batches. So once the qualifications are ensured through a prerequisite checklist which are completely done, then comes the start of the PPQ batches, which we call by definition or a conventional language as the process validation batch. So these process validation batches should be started with appropriate training of all the people. The sampling protocols are extremely important here. So those days we have seen, I'll just take again a minute, maybe a loose talk, but still I thought I'll express. One of the friend, okay, in one of the companies, I'll not tell that, he gave me an example. It's an interesting example, and I'm just, bringing that to a corporate concept or a manufacturing setup concept. In and around Bangalore, I can say, we have these provision stores before this Food World, Spencer's and all these biggies which are come. The provision stores, the Shetty used to be sit in the, Shetty is a owner, we call Shetties in our local language, many of you understand. The Shetty used to sit in the place, there will be assistants for him. Somebody asks something, he'll direct them, and those people will bring. So Shetty is well controlled, he knows in and around of the shop. When Shetty's son sits on in the seat, he is not aware of it. So when a customer comes, do you have Colgate toothpaste? They'll have an assistant. Hey, do you have Colgate toothpaste? He'll ask. That a servant or a worker, he'll tell, yes, we have. So this person will tell to the customer, yes, we have. That person will ask, how much it is? So this person again will ask, how much it is? Then that fellow will say 35 rupees. So this fellow will tell to the customer 35 rupees. Next question will come, how many grams it is? Hey, how many grams it is, he'll ask. So he'll say, it is 250 grams. Now he'll say 250 grams. So what is this conversation we are doing here? Just as a silly example, this is a knowledge transfer what we talk. Knowledge here. Correct? There's nothing knowledge. It's just passing on the information. So if you really see in the process validation, we need people who understand why this sampling design is extremely important. You cannot afford to do mistakes in the sampling protocol. Suppose you sit here in the office and say, under 10 points, you just take a sample from this place, this place, that fellow will be confused. So that's why we say there has to be a pictorial representation. That should be designed in the protocol. That should be photographed. That should be displayed during the sampling approach. That is a level of variability reduction in the design of a documentation. If you look at an industry, again, I can make a very vague comment, okay, not to hurt anybody. The speed of operations and the speed of documentation doesn't tally. The speed of operations is one direction. The speed of documentation is like a tortoise. And the speed of operations is like a hare or a rabbit. Correct? But the speed of documentation is like a tortoise. So this disconnect creates a lot of issues in the organizations. So then the sampling, the details of testing. There's another story to tell again. There is another story which I can say. There is a Kerala person, okay? He is doing his father's rituals. His father saw the cat at the home, always disturbing during the rituals of his father. So he used to tie a cat to the window there. Then he used to perform the rituals. Over a period of time, this person dies. So his son, who is small, keeps observing, okay, when my father is doing my father's ritual, my grandfather's ritual, he is tying a cat. So now, father is no more. No, he has to conduct the rituals. Every day what he used to, every year when he is doing rituals, he'll search for a cat first. So he brings the cat, ties to the window, and then performs the ritual. So what is this example all about? Today, if you see the industry, we know what has to be done. We as people, we do not know why it is required. There is a difference between what and why. So I'm just giving this example on a lighter side. Many of the aspects, we need to know why this is being done. Correct? The moment this why comes into picture, the knowledge, the understanding of whatever activity, what we do, right from warehouse, manufacturing, analytical, or even the distribution. A lot of questions came on the distribution today. So even distribution is also a part of the GDP practices. So the why element has to be part of the documentation. Why the sampling is required, the sampling approach, the rationale of sampling, the justification of sampling should be part of the documentation. Then, if all these things are, especially with respect to the various stages, we'll touch upon the various parameters also. So the design of a protocol, I will not touch upon. The people names have to be clear so that there is consistency. The sampling plan should be clear. What are the tests to be done? What are the specifications? What are the method of analysis? Everything should be part of our protocol design. So if you see the final report, it should also talk about the risk document, the final challenges which are faced. Are there any overways which have happened? Are there any deviations which are there? Are the deviations addressed as per the SOPs? Are the root cause analysis for the deviations or the overways addressed properly? So what are the relevant CAPAs which are part of this investigation so that the future batches are protected? Those things have to be part of the qualification report. If you look at the methodology, again I've just put some picture over there, may not be a right picture, but still as a concept. So it has to be more pictorial in all these validation batches to avoid human errors. Okay, today if you see the industry, whether it is an investigation from OOS or a deviation or a OOTs, so if you really see almost 40–50% of the errors are because of human errors. So we may have the best of the instruments and the equipments, human errors are still happening in the industry to a level of 40% approximately. So again, as Harish told, I've also taken a tablet product at various stages like dry mixing, what are the key control variables within the equipment, how the critical parameters have to be controlled. So if it is dry mixing, the mixing time and the speed becomes extremely important. How do we narrow down in the CPPs? How it is designed in batch record is part of the batch record system. Same thing for binder preparation, same thing for kneading, drying, lubrication, compression and coating. So I've just taken an example of all the critical parameters. But does the process validation stop here? The packaging part also comes into the discussion. If you look at a bottle product, for example, let us take just for discussion on a metformin. It has various counts into the market. The quantity of the sachet to be added into the void space, that becomes extremely important. So what's the void space? How much of the desiccant has to be added? What are the studies required for that? Because if you see the stability data, some counts passes, some counts fails in the market. So what does that indicate? So the packaging studies also are extremely important before we design the packaging configuration also. So apart from the process validation for the manufacturing process, the packaging validation also becomes extremely important. So continued process verification, which we spoke about the stage 3, is all about the real-time monitoring. So there are companies who have gone into high-level statistical tools. Every batch, all the CPPs and CQs are fed into the system. So entire variability, a continuous statistical data points are generated after every batch. The decisions on batch release is taken only after thorough evaluation of these particular data points. So this is one example just to talk about the CPK in terms of the lower graph you see. This talks about the process capability. From a statistical approach, any statistical tool which gives a CPK of 1.33 and above is supposed to be a good data point for that process parameters. Any CPK less than say 1.0, the process needs to go for a particular improvement. So that's the indication of the CPK values. So we also see in the sampling methodology, for example, so how the variability within the blend uniformity is. So when the blend uniformity spec is 90 to 110, all the 10 results are passing. But when it is passing at a variability which is high. So what is the variability in terms of these 10 data points, in terms of a blend uniformity or in terms of a content uniformity is also coming out of the statistical tool over here. So the same things are summarized here, just to tell you on the process validation. Process validation is not only the 3 batch which we talk about. So the guidance is right from the development, then also the prospective validation goes to the stage 3, which is a CPV. So we also touched upon morning, I think Harish touched upon the revalidation. So once we do the validation, any change in the starting materials, be it an API, be it a critical excipient, again it goes for a revalidation of the process. If the batch size from 600 kgs goes to 6,000 kgs, we are adding another variability. That's a big variability actually. For that, again, it goes for another set of revalidation. So any change in the process, again, you want to change some granulation time, you want to change blending time, you want to change, for example, everything goes for a revalidation. And any change in the packaging operations, for example, that also goes for a revalidation. So any revalidation, again, those batches are based on stability as a part of SUPAC guidelines and also the change management process. So the success of process validation, it all depends on the information and knowledge of the product. Today's topic is all about knowledge of product and process. Controlling the manufacturing process to get the specified quality attributes. So understanding the sources of variation, this is extremely important. How do we avoid variability is what everyday thought process has to be there, as we discussed on the YouTube of Rajiv Bajaj. So the understanding the impact of variation on the process and ultimately on the product attributes and controlling the variation. This again, though we understand, we have to control the variability and bring the process into a state of control. So if you look at the FDA observations, I have just summarized in my own language. I didn't want to use that. So there are multiple observations on process validations. That means the way we understand on process validation and the way FDA understands or FDA expects, there will be a wide gap. So there are a lot of gaps in terms of investigations not adequately closed in the process validation. The OOS or the OOTs or the deviations are not addressed properly. The understanding of the operating range of a qualified equipment not considered in the process validation are FDA observations, and this continues to be there in the industry. So there are certain references which are also there. I think the presentation will be shared. So I don't want to take much time here. I think we have covered about the design space. We have covered about the PPQ. We also covered about the continuous process verification. So the next part in the whole chain is on the APQ. And again, the recommendations of APQ are again coming into the same chain is for continuous improvement. So that's it. I'll end my talk here. Thank you.
Sir, that was fantastic, isn't it? The perfect postprandial talk. There was so much of humor, wit, and great knowledge. In fact, I got information from Dr. Girish Pai. He's a very quiet, understated guy. Girish Pai informed me and sent me the photograph also. He is the winner of the first National CGMP Day Award. So please do give him a big hand. So that is, and many people have joined online also, some very, like Dr. Girish Pai, then Dr. Bambulkar Sahib. So many dignitaries have joined the online platform. And thank you for joining. And I now request Dr. Naveen sir, Sharma ji, please, Naresh, Dr. Naresh Sharma sir, please do come over and kindly hand over the memento and the certificate to Mr. Prashant. Please. There's more to come. That was very good. Congratulations, Prashant. It was super. Very good. So now I've got both good news and bad news for you. The first thing, I will go with the bad news. The bad news is we won't have a tea break. So we are going to end with the tea break. After the vote of thanks, you will have a tea break. So that is the bad news. But I'll give you a good news. The good news is I'm going to do a refreshing activity for you. So if you are feeling sleepy, your sleep will get knocked off. So can I request you all to please stand up? Yeah, please stand up. Okay. After you have stood up, please don't look at your neighbors. Don't look at your neighbors, all right? Because you will feel embarrassed. Please raise your hand, both hands, okay? And follow what I'm doing without fail, all right? Follow what I'm doing. Ha, 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 ha. One more round. Ha, 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 ha. Well, that was a refreshing laughter exercise. So much better, and you'll prepare yourself for the next activity. So the next presentation is from Mr. Mahesh Vyas. Please do come over, Mr. Mahesh Vyas. He is a Master of Pharmaceutical Sciences with rich experience in pharmaceutical industries in India, USA in various functions like formulation development, analytical development, technology transfer, QA, audit and compliance, quality risk management, investigations. He has worked as an intern pharmacist in US Rite Aid, in-depth knowledge of domestic and international regulatory requirements which include for UK, MHRA, US FDA, WHO, GMP Schedule M, owing to hands-on experience in governing several manufacturing sites of world's leading multinational organizations. The areas of expertise include QMS compliance, quality metrics reporting, quality audits, technology transfer, supply vendor quality management, root cause investigations. Now that was what Dr. Ishwar sir was emphasizing. Quality risk management, his educational qualifications. He has Master of Pharmacy from Nagpur University with the gold medal, and clap please, and GATE qualified 85.95%. He started with a D-Pharm, very good, D-Pharm, B-Pharm, M-Pharm, and then a foreign pharmacy graduate equivalency certificate with 96.0 percentile, and he's pursuing a PhD at Nagpur University on nitrosamines. So you can get a lot of interesting insights on that. So he has worked, or currently is working as GM QA Quality Risk Management at Strides. So he has done rapid strides in his life. And we have his work at KA GM Quality Assurance, Supplier Quality, and several other employers, which include Glenmark, Johnson & Johnson, GlaxoSmithKline, and Dr. Reddy Labs. So over to you, sir. Fantastic resume. All the best. Thank you very much.
Moving mic?
Yeah, we talked about it. So he will be giving you a hand mic.
Oh, you have.
Hand mic से काम
ठीक है,
करो.
चलेगा. Okay. Yeah. Am I audible? Yeah. Okay. A very good afternoon to everyone. Yeah.
Hey.
Thank you. So today I feel a bit intimidated. You know? I mean, there were giants who spoke before me, and I really thank CDSCO, IDMA, and KDPMA for giving me this opportunity to talk in front of you. And this would be the last session, right? And then one important thing when you present at last is that many of the slides you can say that it has already been spoken about. So I'll just flip over. Anyway, the topic given to me is this product quality review as per Schedule M. Now, before we go into what does it really mean, what is product quality review, I just want to ask you one question. How many of you celebrate 31st December? Can you put your hands up? Right? In fact, everyone. Now, for this year, what we do is, this year on 31st December afternoon, what we do, we sit together for an hour with our family. And we discuss what has gone right last year, I mean the year 2024, what has gone wrong. What are the learnings that you had, right? What are the good things that you had? What were the things that went very well? What were the things that didn't go that well, right? You will realize, and when you have data, you would see the lady of a household and a housemaker, they would note a lot of things on the calendar. When you have that kind of data also, you can analyze the data. You would see that the maid who took a leave in the month of February because her mother-in-law passed away, again took a leave in the month of October because mother-in-law passed away, right? So now what happens? You know, when you sit together, you ponder over, you contemplate, you know, you would decide what has gone right for you, what has gone wrong for you. And that is what precisely, when you do it with product, when you sit together, when you document, that is nothing but a PQR, a product quality review. Obviously, when you do this, your 2025 would be better than 2024. How many of you agree to that? That an hour spent on that, right, would really help you to learn from your mistakes, right? And I have said this in my last talk, and that we are in a business of human lives, right? We are not allowed to learn from our mistakes. Now, we need to have our systems which are mistake-proof, right? We cannot say sorry, right? We probably aeronautical, automobile industry, we cannot say sorry, right? Because we are dealing with lives of people. Maybe if you are in jeans manufacturing factory, right, you can say, even a ton of gunshot jeans, you can sell it, but we cannot do that. And that's how privileged we are. I mean, I always take a lot of pride in saying that we are very selected few, sent by God himself, right, to do this noble job of making people's life better, making their life healthier, right? Now, with that thought, if you do this product quality review, it will help you, right? And any quality system, and I have this firm belief that anything that is doable, anything that is easy, right? That is the one that will anything that would make your life easy will, at the end of the day, would survive. I come from Nagpur, and I was driving with my son, and we were in an AC car, and he just peeped out window. He said, "Papa, why don't people stop at the red signal? They are talking about 45 degrees centigrade, right? Now, at 45 degrees centigrade, if you want people to stop, obey the signal, you are asking a bit too much. There is some innovation done by someone. They have put one green net at the signal, the red signal, and people are stopping there. I heard in Ahmedabad they have done an experiment where, they are putting that water mist also, in addition to that green net. And then you have seen 100% compliance. So compliance, you have to make people's life easy. Understand from their perspective. If things are easy, then only people will follow. Any quality system, if it makes people's life difficult, is not implementable. It will, and that's the lesson that I want to be given in this presentation today. So what are the contents of my presentation? I have what is the regulatory requirement? So I take this, yeah. So what are the regulatory requirements? What is the object? What is the scope and the frequency of the PQR? We will dwell into the different aspect and parameter for product quality review, especially for the finished product. What are the things to be done? We'll see the aspect and parameter for a PQR or product quality review for API. Now it is a regulatory requirement. It has come under revised Schedule M. A regular, and these are the exact wording given in the Act, a regular, periodic, or a rolling quality review of all pharmaceutical products, including products for export only, shall be conducted. So regular, everybody is aware what is regular, consistent. Periodic, with a certain frequency. So PQR, sometimes people use annual product quality review also. It's the same one and the same thing. If you do, it has to be done annually, that's why annual product quality review. What do you mean by rolling? Can anyone attempt? What is rolling? You would have heard rolling plan. Yeah. Thank you, sir. Absolutely, yeah. Very right. So when you have done it last year and you are doing it this year also, you are comparing it with the last year target, you are increasing or decreasing the target, that's what your rolling is. So a regular, periodic or a rolling quality review of all pharmaceutical product, including product for export only. So if you say that I manufacture this product and I am not marketing it in India, but I send it outside, you still have to do it. Where is it mentioned in the Act, right? So it's in part 1, under good manufacturing practices for pharmaceutical products. Under quality risk management, this section of product quality review has been included. Under part 12, that is for API, a specific requirement for manufacture of active pharmaceutical ingredients. Under quality management, a product quality review of an API is mentioned. This is under the revised Schedule M. What are the objectives of a PQR? Now, we have been talking about this particular topic since morning, to verify the consistency of the execution process. When you tabulate, when you have data for a year long, for example, you tabulate simple factors like assay or impurity and those kind of things, you would come to know how consistent your process is delivering, right? So that's one of the objectives. You will know whether your processes are consistent or inconsistent, and Prashant has a lot to say about that already. To verify the appropriateness of the current specification, this is a place where you will, in the PQR, you will tabulate, you will study the specification limit and whether your limits are appropriate or not. Whether you have to tighten the limit, whether you have to, in certain cases, loosen the limit, right? That you would be able to do when you do this annual product quality review or product quality review. To highlight any trend, right? So trend, because you will tabulate data all year, so that would help you to do a trend, right? And then, I was talking on this topic a few months back, and someone asked me that if I am doing each batch, I'm recording the data, I'm putting the corrective and preventive actions, I'm writing everything, then why I need to trend them? I'm doing right first time, then why again there is a of everything, right? I do it right first time, I completely close my kappa. Then again in the year end, why I need to trend them? Now, there's a saying that when it happens for the first time, it's an accident. Right? When it happens second time, it is a coincidence. And when it happens a third time, there's a pattern. Right? And trend is nothing but a pattern. So imagine a situation. You had a very rough day at office, and you came back home, and your wife says, I have a headache. Let's go outside. Let's have dinner outside. Okay. It does happen, right? Headaches, very common. So, yes, you go and have dinner outside. 15, 20 days later, you come in the same manner, battling the Bangalore traffic, come back, and there's another headache. She says, I have a headache. Eat outside. Okay, let's go. It happens third time, right? It happens third time also, right? There's a headache. Now there's a pattern, right? That all headaches are followed by a dinner outside, right? fourth time when you call from office, right? Imagine that you are calling from office. You say, honey, what's there for dinner today? And she says, oh, I have a headache. What you'll do? You buy a crocin tablet while coming, give it to her, right? And say, yeah, have this crocin, let's wait for half an hour and eat at home. Right? So that is what is the kappa that you have put looking at the trend. I'm sorry, Mudassar, I still say crocin. I should have said dolo, but also I'm an ex-GSK, so yeah. So when you look at the data, you identify product and process improvement, right? There are a lot of opportunity that this PQR gives you to improve upon, right? When you look at them, as the example that I have given you at the start of my presentation, that when you sit together, it's very important. All of these factors are important. Sitting together, doing it together is important. Having data, I said that your wife would be, or your mother would be noting down when the maid took leave, right, on the calendar. That is the data. You need to have certain prerequisite in order to come and do a proper PR. When you do that, then there are a lot of opportunity for you to do a lot of product and process improvements. The scope, as per revised Schedule M, it is API and finished product both. Both manufacturer of finished product as well as API have to do, because it's in Part 1 and Part 12. Frequency, as the name suggests, annually. Now, this was around 10 years back when I was handling GSK, and my CMO was Chemwell Biopharma, and this question came that how to do an annual product quality review for so many products, right? And so if you consider annual January to December, it's not that way. You can do it from the time the product has first come into the factory. So suppose you have a product launch. You have launched a product in the month of May. Suppose you have launched a product in the month of May. So you can take a data from this May 2024 to May 2025. It is still an annual product quality review. Right? Annual does not mean January to December. I have seen many people, they have a product, they will do an APQR. Suppose they have a product in month of November, they will do it for a couple of months. Right? That's not a great idea. You will not have enough data to evaluate. So, you can do November to November. It's not been said anywhere that an annual product quality review has to be from January to December. Right. Now let's see what are the aspects and different parameters for product quality review. So review of. It includes review of starting material and packaging material. It includes a review of in-process control and finished product results. These all are given in this revised Schedule M, right? All of these are given very clearly. So, and the Act very clearly says that minimum are the things to be done. You can include new things also, but these are the minimum requirement. These sections have to be there in your APQR. A review of batches that failed to meet specification and their investigation. So you need to, we will go into each and every point in the subsequent slides. A review of deviations or non-conformity. A review of changes to process and analytical methods. A review of dossier variation and post-marketing commitment for new dossiers. A review of stability monitoring program. A review of quality-related returns, complaints and recall. Review of CAPA. Qualification status of equipment and utilities. Last but not least, the technical agreement. So total, I think 11. There are these 11 sections under which you have to do your product quality review. And I think I mean, most of us are doing that, whoever who is doing product quality review, but this is the minimum requirement. You can add new things. You can add a whole lot of the process capability things also, right? In your, a lot of people do, and what Prashant has just said, the continuous process verification related stuff also you can add to the, so there is no limit to data that you can review. Obviously, you need to have a data to review first, right? A meaningful data to review. Let's talk about starting material and packaging material. That's the first thing. Now, the points in the inverted commas are just taken like that from the Act. Review of starting material and packaging material used for product, especially those from new sources, and in particular the review of supply chain traceability of active substances. So you have to do a review for all your starting material, API, excipients, packaging material, and it is specially written that you need to review those material, those are from new sources. So that has to be specifically included, a material coming from new sources. Supply chain traceability, right? So who is the original manufacturer of the material, right? Who are the distributor? Who are the repackager, right? You should know exactly what happens in your supply chain. So the complete supply chain traceability for an active substance has to be there. I mean, you would be seeing that, a lot of these manufacturers who manufactures a bigger batch, and then there's a repackager who would repack them into a smaller container. That repacking is also a GMP activity, right, happening in a GMP environment. And hence you must have a supply chain traceability. That is what's been said. You have to train the critical material attributes, the identification. A lot of these has been already spoken about. Identification, assay, related substance, particle size distribution, all of them needs to be tabulated. I have just given you one example of assay, how you can do it, but the same can be done for all the critical material attributes. There will be a lot of learning when you have tabulated this, right? For example, an assay of an API and a corresponding assay of a finished product, that same batch, you would come to know that whether your assay value is failing because your input material assay itself was lower or higher, right? So a lot of things can be, you would come to know by doing this. So that's on starting material. Review of in-process control and finished product results. A review of critical in-process CPPs, in-process control and finished product results, CQA. And I will not go into detail all of, though I have an attachment, which I can open. But all of these have already been spoken about. Your mixing time, your granulation time, your pre-compression forces are all critical process parameters. If you maintain that, you will have a good critical quality attributes. Right? So assay and all that. So a review of all this, and you can take directly this data if you are doing, continuous quality improvement programs and, CPVs and all that. Review of batches that failed to meet specification and their investigation, right? So now you have done investigation already, you have closed them, but when you do them, do tabulate, I've just given you one example that how to tabulate, what are minimum details that would be needed. But when you do tabulate them together, when you review at them, you look at the kappas together, it may give you a new signal. And you can learn from that, So maybe in our industry where there is a lot of attrition, people leaving, you may not be knowing. And what happens, as Prashant also said, that, with people, the knowledge also goes, right? But the best thing to retain knowledge is to retain a good document. So if you have this, you know that what are the kappas that were already taken, what is that is working for us, what is that's not working for us, right? So a review at the end of the day, at the end of the year in case of an APQR, will help you to design some new kappa if at all that is needed. Deviations, non-conformity, again very closely related to investigations. A review of this is what has been written in the Act. a review of all significant deviation or conformity. There, it's very clearly written significant. So for our common understanding, let's talk about major and the critical deviation at least. So you should review them. And the effectiveness and the resultant corrective and preventive actions taken. So any corrective and preventive actions that are taken because of your deviation and non-conformity, that needs to be tabulated. Review of changes to the process and analytical methods, dossier variation and post-marketing commitment for the new dossier. You know. So again, the word from the Act, a review of all changes made into the process or analytical method, a review of dossier verification submitted, granted or refused, post-marketing commitment for the new dossier and the variation to the dossier. All of them you can tabulate together, you know, in the manner that I have just given you one example. You can write the change control number. That's how you, I mean, change control, again, one area that a lot of us in an APQR would review what are the changes that happen. But these are change control that are very, very specific to the variations that we file. So change control number, the changes that are incorporated, what was the category, what was the justification for that change, what is the implementation status, what is the regulatory status, have you filed it, you know, whether you have got the approval and so on. And the one that is pending obviously will not have an implementation date. So you can file, you would know that what has been filed. This will help you to plan the next year batches also. Right. Review of the stability monitoring program. Right. A review of results of the stability monitoring program and any adverse trend that you can see. I mean, you would see anyway in a good organization, you would keep on looking at the trend, you will keep on punching the data in whatever software you use in an Excel sheet, and you would, obviously it is required to look at the stability data as and when it comes, you know, not at the end of the year. But if you are not doing it, at least at the end of the year, you would come to know that what is my trend, where is my product going, what actions I have to take right away, right now. You know, when one batch is cooking in stability, you may have another batch with the same formulation that's going on the shop floor. What are the actions taken for already existing batches? That's what you can do when you review your stability data. So that's a requirement now. Till now, this was good to do. It is now very must to do. Review of quality-related returns. What is return? Quality return? Return and a recall. What is the difference between return and a recall? Anyone? Mm-hmm. 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 Yeah. Yeah. Most of you are very right. So return is in your control when it is a voluntary decision by you, which may not involve patient safety and efficacy directly. When a regulatory body is not involved in all this, return is your own choice. For example, you have sent a consignment outside, and the truck unfortunately met with an accident, and you think there will be a damage to the product. You are asking the truck to come back, right? Or a lot of these products that get expired in the pharmacy, at the medical store, you know, so you have to return them, right? So they may or may not have any effect on the consumer, but recalls are different. Recalls, if there's a regulatory body involved, you know, there is a suspected adverse event that can happen to a consumer or a patient, right? So a review of quality-related returns, complaint, and recalls, and the investigation performed at that time, that needs to be reviewed. What are the different areas in this particular case that you would review? You would review the batch numbers, the complaint, what was the complaint number, confirm whether the complaint was confirmed or not. What was the complaint classification, the status, investigation summary, and outcome. I mean, I'm very passionate about the subject of complaint, actually. You know, one complaint, a lot of time when we see, people say that only one complaint is what I have received. right? So if you have received a one complaint, that means there are 1,000 of those more which are there but were not able to complain at all, right? So one complaint, or many a times people say that I will wait for certain more time, I will wait for another 2-3 complaint and then I will investigate. This just one, right? And if you see, if you really see how many times we complain, there are a lot of opportunities that you get. You normally do not complain, right? When your day starts with, say, brushing your teeth, so it starts with, say, Colgate in the morning, right? That's the first thing that you touch. And this was long back, around 15 years back, when I had that first Colgate toothpaste in my hand, and I saw it's written there that calcium carbonate, which will make my teeth stronger, right? So, toothpaste, so you brush, right? You brush, you have everything in your mouth, you spit it out, and that will make your teeth stronger, right? As if you have taken it inside, and then you'll say, yes. Who does that, right? Nobody, everyone spits it out. Is there anyone in the room who wants to make their teeth stronger, right? But it's, I felt like complaining, right? This is not the way. Calcium carbonate, the bioavailability of it, as a salt itself is 50%. The calcium loading itself is 40%. So it doesn't, the bugger doesn't get absorbed when you take it through oral tablet, and you're talking about through a toothpaste, right? Then you feel that brush has a problem. This is not as soft as what it was advertised. You go for a bath, and there's a shampoo, right? And this shampoo, Katrina Kaif says that the hair will be like her, it's a very silky. And I've also used it many a times. Now, my hairs have not become like Katrina. Actually, it has become like Mohammed Kaif, So many times I feel like complaining, I haven't done that. Right? Then you have to go catch a flight. There's this web check-in. You will not get a window seat till you pay for it. Right? Have you, anyone, great Paramvir Chakra holder, who have got a free window seat in Indigo? No, you'll not get it. Though, you got it? Oh man, you are a Paramvir Chakra anyway. You go to airport and there's a whole chaos there. A lot of things to complain about, right? And your life is just like that, but we hardly complain. The point I want to make is that we hardly complain. So if at all there is one brave person who is complaining, he's really, really brave. He is someone who wants you to make a serious change to the product. That's why do not worry. Even if it is one complaint, you trend it. You see how it is, what is the kappa, whether the actions that you have taken are sufficient, whether you need to look at them again, and so on and so forth. Review of the kappa we have already discussed. Review of the qualification status of equipment and utilities. That's another section that has been there, right? The qualification status of the relevant equipment, utilities, heating, ventilation, air conditioning, water, compressed gases, and review of result of monitoring output of such equipment and utilities. So not only their qualification status, but also the parameters, viable, non-viable count, differential pressure, air velocity, whatnot. You can trend them, you can review them, you can see whether you are under-validating, whether you are over-validating, and all that. What are the lessons that learn? You can correlate these qualifications with the type of complaints that you are getting or type of deviations that you have, and see whether there is a correlation that exists between your equipment qualification and the complaints or a deviation coming out of that equipment, right? When you look at all these data together, that is the time you will have certain new insights, certain new things, and that is what you need to act upon. Because this is the first time probably you are looking at everything together, and that's how a PKR, good PKR process helps you. Technical agreement, last but not least. You need to review your technical agreements, whether they are relevant to the date from the last time when the technical agreement were done, whether the new products have been added, whether the new clauses have been added. This has happened during the time when that all concept of heat-treated pellets came into existence, right? And from the time, and many a times, what happens that you start revising your technical agreement from the time the new guidance has come in place or new requirement has come in place. For example, the DEG, EG requirement and all that, right? So you will start doing it from the time but you will forget to do it for the products that were done before. So a review of all technical agreement together would help you that, okay, we have added some product. After this guidance, we have not added that particular clause in this agreement, and why not to add the clause? At the end of the day, when you do it properly, it has to improve your life. It has to improve your quality of life. Then and then only it will survive, right? Okay, so once you do all that, then this is the most important part of a PQR. A PQR must have a good recommendation, And I will borrow a line from my friend, my guide, my mentor, Prashant here. He used to say always, operation successful, patient dead, right? Many a times we do our documents like that, APQR done, everything done, all sections included, no recommendations coming out of it. It's waste of time, energy, exercise, don't do it. If you have to do it like that, don't do it. But if you are doing it, you must learn from it, right? Don't be in that situation that, you have spent so much time, because this is a very time-consuming activity, right? And there's a purpose behind asking you to do this, that you must learn from your mistakes. You must learn. You should continue to do good things which you are doing already, right? And hence, this section is most important in my opinion. Whether the CAPA were adequate, right? Whatever, just think about it. For all the companies, all non-conformities, deviation and all, whether those were adequate, whether you need to put new actions, right? Whether validation, qualification are adequate, right? Whether revalidation need to be performed. There were different things that were said for the revalidation, under what condition revalidation has to be performed. So whether that needs to be done, right? Based on the data that you have in certain equipment for certain process, facilities, whether there's a need to do a revalidation, that you will only come to know when you have all this data together. There shall be procedures for the periodic management review of the action arising out of PQR. Just read it. I'll give you 5 seconds. What does it mean? Whatever are the actions that are coming out of periodic product quality review, there has to be a management review of that action. That means, in your management review meetings, monthly meetings, quality system review meeting, whatever name you suggest, the actions that are coming out of a PQR should be very clearly mentioned there, and a plan, what are you doing on that? What is the progress that you have on these actions must be documented. It must be told to the management, what are you doing, right? Now, review of these actions that are coming out of a product quality review, that should be verified during your self-inspection. So it's not that you only review, your self-inspection, internal audit, whatever systems you have, how have you done on the product quality review related actions? Are you implementing or not? That needs to be checked during self-inspection. These are the things that are written in Schedule M, the revised one. Nothing is my own words. Nothing is my own whims and fancy. It's all written. Are the requirements now. Earlier, as I said, it was good to do. Now it is must to do, and you'll eat the fruits, right? Once you do it for a year and a half, couple of years, and you will start seeing that there is a lot of advantage in doing it. The responsibility of conducting a PQR and implementing the actions should be defined in the quality element. That is also has been said. that in your quality agreement, suppose you are doing a quality agreement with a CMO, who is the person who is responsible for doing a doing a PQR or an annual product quality review shall be defined. Right. Last but not least, the authorized person responsible for final batch certification. Right? The authorized person that we have as per the Act, as per the Schedule M, that person shall ensure that the quality review is performed in a timely manner and accurate. So it's a very clearly the responsibility has been assigned to a single person. It is the responsibility of an authorized person, a certified person, to make sure that a PQR is done every year. Right? So if it doesn't happen, you are the one who will be held responsible for that. It's very clearly written. Okay, aspect parameter for product quality review. For API, I will not go into the detail. Those were 11. Here there are, I think, 6. So we have already covered that. Only you look at them from an API angle. Right, so for API under part 12, in-process control and finished product results is what, as an API manufacturer, you have to do. You have to do a review of batches that failed to meet specification, their investigation, critical deviation or non-conformity. Changes to the process and analytical method, the review of the change control. Stability monitoring program for your API. Quality-related returns, complaint and recall. Adequacy of CAPA. These are things that needs to be done. So thank you very much for your patient listening.
Thank you, Mahesh Vyasji. I think very, very detailed PQR. I think personally what I learned today, I think there's been a kind of a great learning experience for me as well. And I think one observation, the paracetamol, the favorite is not my monopoly. I think after me, Prashant, as well as now Mahesh, the favorite example has been paracetamol. And paracetamol is always a learning experience, as Dr. Ishwar Reddy ji also said, that most of the time the dissolution failure happens only in paracetamol. Not only with, I think most of you who have been, I myself, when I was working on shop floor, I would have made thousands of batches. But I'll not be sure whether can I make a batch without a capping or a sticking, or a friability failure. So these are the kind of variables what you have in the tablet manufacturing. I think, thank you Mahesh Vyasji for giving us such a great insight into the what you call as a product quality review. Now I invite Shri Rajshekhar ji, our Deputy Drugs Controller India, CDS, kindly honor Mahesh Vyasji by way of a certificate of appreciation and memento. Thank you, Rajshekhar ji. With this, it's not the end, it's a beginning. I think that now coming to a beginning of the most exciting part of today's event. I invite Shri S.M. Mudaji to initiate the panel discussion. Sir, it's all yours. S.M. Mudaji will moderate a panel discussion and Q&A session.
Yeah, hello, good evening. I think there are a lot of questions to be asked, and we intentionally kept sufficient time by avoiding the tea break to make sure that we make an attempt to answer all the questions that we have. Very interesting day, lot of good learnings with practical examples, and we thought we should do. And I would invite all the speakers to come on the dais so that we can start taking questions on their topic. So may I request, starting with Ishwar Reddy sir to come on the dais. Prashant, Mahesh Vyas, Dr. Naresh Sharma, I want you to be here because I learned while I was talking to him privately, he's one of the greatest scientists in product development. He has huge knowledge of what has been spoken in the presentation, and I'm sure he'll be able to give some practical examples of his experience to answer certain questions. Right. Harish, yourself. Yeah, Rashagri sir, please. Yeah. Right. So how do you moderate? You have questions from people. So you can ask question directly by taking the mic. If you want to write and send the questions, you can do that. Meantime, I have a few questions which have been received online. So it has been sent to me on WhatsApp. So I'll be asking those questions as the fillers. But in the meantime, so let us start. Anybody has any questions? But before that, Prashant, to answer his question, how do we interview a person? I think you made a comment that people can pass through good interviews and they can get into the job, but what they know is a different thing. So I can tell you my example of one such interview I conducted. I received a CV of a R&D gentleman, and he had put in the list of products he's developed, almost all products of the country. In that, there were some very important formulations, difficult to handle formulations, which we knew. He had put all of that, and with a lot of confidence, he said, I would like to come and meet you. So I called him. First, I had a phone call, then I called him. But before he came, I had done something that he did not know. I purchased some 6 key products, which I knew were difficult to develop, market products of the company for which he marketed those products and kept in my drawer. So when the interview started, I started opening one pack and started showing to him. So none of the products were stable. So I think that's what we have to do, not only by asking questions in the interview, but demonstrating with examples. So anyway, so let's start. Any questions from anybody?
Oh, sir, my question to Ishwar Reddy sir.
Yeah, can you tell your name and the company? You can stand, please. Yeah.
Myself, Niranjan, sir. My question is to Mr. Ishwar Reddy sir. So now we have to do bioequivalence and bioavailability study by everybody as per schedule revised M. So we are doing the marketing for domestic market. Whether the manufacturer can do such an investment and do bioequivalence study and bioavailability study for products.
What is the question like?
His question is, just let me rephrase your question. I am a small scale company, whether I need to do BA/BE study for the domestic sale of my products, which because of BA/BE studies are very expensive. Is it right, your question?
Correct, sir.
Okay. I think you know the answer also.
Answer.
See, you know the answer, that's why you asked me the question.
Answer we know, but the returns are not that much in the domestic market.
I am not in the trade business. Otherwise, I could have purchased your product and I could have become your distributor. I could have make sure that you will get a profit from that product, provided your product will have complete data and information as per required under the Drugs and Cosmetic Act. So being a drug regulator, unfortunately, the Drugs and Cosmetic Act does not differentiate the size of the industry. I think you are aware. Irrespective of whatever your size, in fact, that's the Act does not, even our rules, regulations, nowhere stated, differentiated the size of the company. So for me, all are equal. And these regulations are applicable to everyone. But whatever the question you asked, it is a very genuine question. because spending almost more than 10-12 lakhs rupees for one product is not a simple task for the everyone. That is the reason in the morning, in my first opening talks, I said, you reduce your burden from your shoulder. Major problem with you means so people are not affordable. I'm talking about them. Those who are not able to affordable them. On your shoulder you are weighing so much of weight. Weight means number of products. You see how many products license you are holding. That is actually feeling heavy on you. You discard as much as possible which you are not manufacturing or which are not applicable for some products which you may be manufacturing once in a blue moon. And why you are keeping so much of load on you? Be relaxed first. Whatever your major sales, your bread and butter, you identify your product, you focus on that. Then definitely you have reasonable money to have good data on that particular products. Otherwise, if you see basket, I don't know, but you may also have more than 100 products in your basket. Okay, so which will be very difficult to have the complete information and data. At least my suggestion to you is, whatever products you are going now onwards, be very cautious. Unfortunately, what happened, whatever I'll have 25 years of experience in the drug regulation, most of the manufacturers, they will burn their hands by looking at the others. If any one particular molecule is moving little better in the initial stages, everybody will jump on that molecule. And that molecule will get collapsed after some time. The moment, the day you entered, it may be, for example, the market it is launched, it is 100 rupees. You thought that even if it will come down to 50 rupees also is beneficial, it is a profit for you. With a 50 rupees intention, you enter into the market. By the time you enter, that 100 rupees will come to the 20 rupees. So I think you have to see the market dynamics also. And first of all, first you should see really how much capacity or competency you have to manufacture that product.
Or else can we revise the price as per this revised Schedule M?
Revise the price. BAB study is price is not regulated by the government.
Or else we have to manufacture only one product by one manufacturer.
Not a one. I'm not saying one product. What I am trying to say that you know what is your strength. See, for example, if you have 5-6 dosage forms, out of that you have very strong in, for example, say external preparations like ointment, creams.
Yes,
you focus, you expose yourself, put all your efforts in that one dosage form and have reasonable multiple products in that area.
But
So that is what I'm trying. Of course, it is very easy for me to say this. When it comes for your implementation, you have to, think very, open with open mind so that you are able to take a proper decision. I agree with you. That's why, yes, it is expensive process, and because of we never spend 12 lakhs rupees for the one study. That's why, otherwise, those who are regularly doing, for them, in fact, 12 lakhs, I said at the, minimum side. I think it may be sometimes even more than 20. Yeah, even if you do at the USFDA approved facilities, it may be a 25-30 lakhs rupees also.
Okay, sir.
Okay, but you had to do a lot of permutation combinations. You had to take a call how to overcome. But in this government revising the price, because the government is not regulating that prices of the BAB studies. But I'm sure when the demand is more, sometimes the price is automatically comes down.
So when the Schedule M is introduced by government, whether the regulators can instruct the manufacturers to manufacture any one particular product by one company.
But
So
you know one of the major problem what we are receiving? They say that in India, human resource is more expensive nowadays. Can I involve and tell you, reduce your salaries? We got that, one particular QA manager, head QA, corporate QA,
Yes.
and there may be at least 50 lakhs and above package, up to 1 crore, 2 crore packages.
Yes.
But I think
And somebody can come and ask that, sir, put a regulation on this salary also. There, government should not enter into these areas. What I am trying to say that this is actually a business. Wherever business is there, government. See, we are involved. Why we are revising the schedule M? Because of the quality is involved. For further assurance the quality. So wherever quality is there, government is part of that.
Okay, sir.
I
Thank you.
think, sir, yeah, just to add to the point, I heard Dr.
Thank you,
Reddy sir several times earlier on this topic. What he's making a point is that you handle products that you can handle. Even today morning presentation he said, you can't have a laundry list of products that you intend to manufacture, have on the license, and then make an attempt to manufacture. So idea is I'm sure even in the loan license era, we had the list of number of products you can handle based on the capacity. So the capacity that we have and products that you can manufacture has a correlation, and I think it makes sense. So it is not the session to decide business strategy of somebody. What you should do is your call. But ultimately, one thing is very clear: as drug regulators, they have every right to see whether you're capable of doing what you claim you are doing. I think from that standpoint, some kind of control on the numbers and the types of products has to be there if you really want to deliver a good quality product, I think. That's the message. Any other question?
Yes, sir.
Please, sir.
Sir, this question is to Prasant, sir. So myself, Rajendra Kumar Parida, Manager QA Juventus Healthcare Limited. My question is, suppose one product, we have stage, everything, CPP, CQA, everything we have validated in the process design stage. And that product technology transfer we have done to the site, then we have validated one product. And first page, whatever data is there, everything we are getting. So whether we need to confirm another 2 batches or 1 batch data is sufficient.
Now, technically, the PPQ, what we told, as per the guidance, 3 minimum batches have to be established with statistical data also. So 3 batches are minimum required. If you look at the FDA guidance, there is some terminology called concurrent validation, which is not accepted. Wherever there are certain radiopharmaceuticals, as per the guidance I am telling, where the shelf life is very, very short, there are certain products which are limited, where the batches are limited, accepted by FDA, in collaboration and liaising with FDA, those products can be exempted from 3 batches as per the guidance. So technically, to answer your question straight, 3 minimum batches are required to close the PPQ batches.
Okay, one more question.
Yeah.
Suppose that continuous process verification CPV, if you are doing the APQR, so whether it is required to do the separate CPV?
Yeah, APQR contains, as Mahesh Vyas also has briefed, some 11 elements to be considered in the APQR. So if you look at the CPV part, it's more on the CPPs and the CQS. So this is to demonstrate, because your APQR will be as per the annual schedule. So to even the 4th batch, for example, or 5th batch tomorrow, day after tomorrow, 6th batch, I'm just giving an example, it has to be on a continuous basis. So this talks about the CQA and the CPPs.
So that is required.
Yeah, APQR talks about all your deviations, OOS, agreements, as Mahesh briefed, for all the 11 elements.
So what is my doubt? So what are the parameter we are capturing in APQR?
Yeah.
Same parameter also it is there in the
That
CPV.
part of CPP and CQA, once you have done for CPV related project, you can use the data for the APQR, but not vice versa.
Okay, thank you.
Yes, sir. Thank you very much, first of all, to all the speakers here. And I could find out most of the people here with a background of formulation and very few from the API background. There's something to do with the API industry. Sir, internationally, to approve a vendor is a very cumbersome process. It's a process in itself, rather, I should say. It's a process which takes 6 months to 1 year to qualify a vendor. But India, being a price-sensitive market, the vendor can be changed overnight. Do you think after the implementation of revised Schedule M, the changing of the API supplier or API approved vendor, will it going to be that easy or is going to be more difficult, sir? This is, I think, Mr. Vyas, Mr. Prashant, and even on a regulator front, you can also make a statement, sir. I hope I am clear to my question.
Yeah, right. See, the business-wise, once you are tied with an API and have enough time, you can change the vendor. So if you really see, it's like a wedding. I use the word in my local language, it's like a wedding. You are tied up to the API vendor For example, I'm talking from a formulator perspective. The moment the type of data which has to be generated, including the stability, including the vendor qualification, it's a long process and it's a cost-involved process. So as to the extent possible, depending on the business risk which you anticipate, with the risk anticipated with the vendor, it depends on the manufacturer's guts or the risk appetite, whether to continue with one vendor or more vendors. So that is what I feel. But preferably from a business continuity, there should be more than one vendor.
Yeah.
As an API manufacturer, if I need to change my KSM, if I've been supplying my product to a regulated market, I need to do the re-stability studies because I'm not sure the KSM manufacturing process is the same. The impurity profile might get changed. And if their impurity profile gets changed, even my API impurity profile will get changed.
Okay.
So if I'm supplying a product, and there's three more manufacturers supplying the same product, but the impurity profile will be changed.
Yes.
It could be different impurities, different values. So now how that is going to be reacting to when they do the formulation, it might react with the excipients, it might react in the whatever the process solvent they do use, how they can be very sure when they change the source from X to Y.
Yes. So the formulator, there are two aspects of discussion here from what the understanding of the question. The change control of the KSM vendor by the API manufacturer should also be notified to the formulation development or the formulator also, or the formulation manufacturing company. So their quality assessment as the change control assessment will consider your changes of KSM also. And in that documentation of a risk as a part of change control, the studies done by the API manufacturer should be part of their documentation also. That's extremely important. From an API manufacturer perspective, if you see, it becomes extremely important when you change the KSM vendor. As you rightly told, the route of synthesis would be different for the KSMs. The number of solvents which are used will be different. The ability to control the residual solvents in the KSM again will have an impact on the API. The change assessment or the risk assessment by the API manufacturer should consider all these aspects into consideration. Provided if you keep one specification on the API, there also could be additional tests should be incorporated in the API specification also. If a solvent, for example, by KSM vendor 1 is changed, you have to build that as a part of your risk assessment. And if the demonstration is not there in your development, it has to be put into the control strategy. Your residual spec change for the API will change. That again goes into the stability. So all those things has to be considered as a part of extensive change control management. So it's a long process. It's a extensive cost involved project actually. And there may be a spec for API for some of the APIs. I can quote some examples. The impurity profile, you may have to consider all the impurities in your API spec also as an additional test. Otherwise, your formulator will not be accepting. I hope I answered your question.
Or you want to add something? And just to add to what you said, that how difficult and easy it would be to change a vendor overnight, it would not be easy, right? Because only people who have this data, and I have just given you a requirement for a PQR, right? But then there are a lot of new things that API manufacturer have to do now, right? So only those players in the market, certainly you are there, right? So only those players in the market who have ability, appetite to have this data, have that kind of infra, that kind of people would be able to cater to the market, right? So it is going to be not that easy that you just change it with your whims and fancies. It will change now.
Yeah. Sir, in the meantime, I have one question. Sir, you are adding to that?
Yes. The question what you had asked is really very valid question, and is also the need of the day, that we need to understand the basic concept and about that how to have the risk factor consideration while moving ahead. The first thing that during qualification of the vendor, even at the R&D level, one must have 2 to 3 vendor. One is the main and another is the alternative vendor. Other part, while doing the process validation and stability, entire set of both the batches for 3 put into the stability is required. That's what the confidence level need to be built at the manufacturer level. Something has been taught by Prashantji and the other speaker about OVI, organic volatile impurity. Let's not restrict only up to the organic volatile impurity. Just see about the excipients as well. It's not only API what we're going to talk. There are classical examples, for example, like etoposide, which is using that propylene glycol. The change of vendor for the propylene glycol itself will have the dissolution profile and crystal formation in the end product. There is one excipient, citric, which is to be added as a pH adjustment for the etoposide. The entire process what has been discussed throughout the day is very, very valid. All the speakers has elaborated starting from, say, pre-formulation for the product development, then goes to the prototype of formulation, optimization, then technology transfer, troubleshooting. These steps are really very, very important to build the confidence. Coming back to that, which way we have to add the citric acid, again a question mark. The time the PG is going to be changed for the etoposide, if it has not been appropriately developed, appropriately transferred to the production label, and the addition of citric acid before, say, mixing, or at the end while adjusting the pH. These are the very important part of the citric acid need to be added at the time of dissolving the active moiety in order to avoid the crystal formation and to reduce the process time as well as the temperature. It's a big science. I will not elaborate in detail, but this is a classical example. Each and every step what during the process validation presentation has elaborated those two important slides. Critical steps, identification, and how to address those are really very, very valid. So it's not only organic volatile impurity, but we also need to concentrate about each and every critical excipients as well.
Yeah, Dr. Sharma, since you spoke of process optimization, I will
Just
follow.
let me.
Sure.
I see, कभी है ना, those who are able to understand the वो दिल का आवाज सुनता है ना, वही बोल क्या आंसर है इसका. I know, being a API manufacturer, you know, today I'm he supplier, next day, but see, end of the day, if we will tighten the system, everybody is strictly implemented, it is actually good for the manufacturer. Means you have a fixed supplier, your vendors. See, initially it will take some time. See, this is like this, you know, don't mind other way, having our relation before the marriage, after the marriage. एक बार शादी हो गया, that relations are strong. Okay, so the way you make the relations with the, he said rightly, it is like a partner, it is a marriage. He said very rightly, it is very difficult to live. Actually, it is one way good for you. Today it may be thinking that, you know, I may not able to get a new formulators for you, but one day you feel that those who are already with you, he is not leaving you. Okay, so don't afraid about your schedule. It is good for you.
Yes, sir. So there is one question for Dr. Naresh Sharma. Are engineering batches, I'll rephrase it, do engineering batches require process optimization? Or the question is, he would like to know whether process optimization batches are actually required before you go for actual process validation.
Well, process optimization during the product development stage or after the technology transfer?
I think during the scale-up stage.
You ask that person who has asked.
No, it's come, sir, online questions are there. Yeah.
So there are two steps what I can understand about the optimization. During the product development, there is a set of trials to be taken up, and there are 60 to 100 trials for the process optimization put for the stability study and the fine-tuning of the formulation done at the research and development level, and further goes to the technology transfer for 3 batches followed by a stability study. But as Prashantji has mentioned that there are the 3 batches, but this is also another part. During the technology transfer, the 100% success rate is not expected, right? There may be some changes while scale up and during the technology But if some variations happens during the technology, then the guidelines clearly say you need to take the number of batches till the confidence level developed and the consistent result produced. It may be 10 batches or so. Then again, the concurrent validation concept was there. Prospective was the initial one, concurrent, and retrospective has been outdated. So concurrent point of view, the guidelines are well accepted. Up to 10 batches you can take up. If still you are not getting the optimized process as well as the expected pre-specified parameter compliant product, then you need to go again back to the research and development and have the again regeneration for the product development and back to the product technology transfer.
Okay, so. Yeah, thanks. I hope.
First preference give the people those.
No, yes, of course, yes, yeah. So I just wanted to know, since the topic was there, optimization, yes. I'm sure people who are online, they are also eagerly waiting to see what is happening here. Yes, question, please.
Yeah, I have a question. Myself, Dayanand, heading Quality Control Department in Juventus Health Limited. I have a question to Dr. Ishwar Reddy, sir. An earlier version of Schedule M was having a requirement of API expiry should not cross, our product expiry should not cross API expiry. Now that clause has been removed from the guidelines. So what is the regulatory requirements from the industry?
Thanks. You are not asking why you have removed this. Otherwise, you ask your KDMPA, write a letter to incorporate back again that provision.
No.
We had a seminar on PIC.
Yeah, that's true.
6 years back, I asked you the same question.
That's true.
So my presentation was there of 2011.
Yeah.
And I'm happy that 2024 is the light of the day.
So if you see our revised schedule, yes, as we have omitted that particular clause, but we have also incorporated the concept of retest date, which was not there earlier. Earlier it was you had to have mandatorily expiry date for the APIs. Retest date is not applicable for the formulations. Please remember, don't extend your formulation expiry date, by again validating, and retest date is applicable only for the API. So that's why when you introduce the concept of retesting, then there is no meaning of having restricting the formulation shelf life with the leftover shelf life of the API. Is it clear?
Yes, sir. Thank you. Respect to the bioequivalence, sir, in continuation to our earlier discussion, can we have in vitro and in vivo correlations instead of bioequivalence for the already existing products?
Even for the biopharmaceutical classification class 2 and 4?
Yes.
You means earlier you did some BAB study or without doing any BAB study?
No, earlier means I'm talking about the product which is already established and it is in the market. And
if it is already in the market, if the product, then there is no need for doing anything.
Okay, as a competitor product to with the innovator
See,
product.
the regulations are always applicable for the perspective, not the retrospective.
Okay.
When any notification regulatory requirements are there, with the day when it is notified, okay, that regulations are effective from that particular day. Or otherwise, if we'll ask them to do everybody BAB study, then whatever BAB centers in India are there not sufficient. We have to hire globally all the BAB centers. Okay, but this is required from the date it is effective. Okay, so if it is already established product, if it is, but yes, if you have changed any major in the formulation, then definitely you need to do.
So can we have in vitro, in vivo correlation study like a competitor?
No, that's why I'm asking you. Try to understand whether you are asking about the product which is already in the market or which are going to be placed in the market.
Already there in the market for some different manufacturer, and I am going to enter into the market.
Oh, that means for you it is a fresh.
Yes.
You tell me that I thought that your product is there in the market.
No, no.
Irrespective of that product is there in the market or not, if you are introducing, if it is that product comes under the biopharmaceutical classification 2 or 4, you have to do BABE. That is a mandatory.
Thank you.
Sir, when I mentioned about in vitro, in vivo correlation in my presentation, this is a step prior to a bioequivalence study so that you can predict what is going to be your bioequivalence and your bioequivalence study doesn't fail.
That is best product, your best formulation. So, while design, you design the product with 3-4 different combinations. Okay, out of this, which is the best? Which release profile of the particular product is the best? That you can take correlation with your BAB data.
Yeah, I have a question. Though US FDA is stress upon the data integrity.
Introduce yourself and your company.
Yes.
My name is Ramesh. I am from Steriscience Specialties Private Limited. Though USDA is stress upon the data integrity issues, still recent inspections we could see the DI issues. Maybe because of this reason, FDA is still continuing the unannounced audits. My question is with the CDSUO, KDPMA, IDMA, is there any micro programs to bring the industries out of this image and make the FDA to visit India with announced audits?
Okay, I will answer on your behalf also. So my dear friend, in the morning I made in the second slide, out of 25 top generic manufacturers in the USA, top 10 are from India. If USA don't have a trust, confidence on you, how can we will be in the top 10? That is the first point. Second point, this surprise audit concept of US FDA, it is not only restricted to India. It is their policy. No regulator policy will be made for country specific. They are not targeting you. So it is a US FDA's policy. Okay, thereby they are doing all this type of surprise inspections, not only in India, wherever they have their approved sites, they are doing everywhere. Okay, so that's why I think it is not I don't think so why you are feeling uncomfortable or image is not good. In fact, other way around, we are proud. Even if you come on surprise inspection, I am ready. That means really you are challenging. Okay, if you want, you can come, no problem. That means I take other way around, really we are proud of our industry. And so in fact, think other way it is a stressful, it is this, it gives more confidence on ourselves, on our systems and our procedures.
Right. I think well said. Basically, it is not the industry or association issue. It is a unit issue. It is a company issue. It is the place. And what we have seen is that India is so vast. Our way we understand GMPs and our approach towards compliance varies from south to north. I have 18 plants who worked from Pondicherry to Sikkim. Not to say that anything is wrong, but then our understanding of what compliance means, what can be accepted and what cannot be accepted, it varies. It is a poor understanding and misguided approach that has led to this. So association cannot do anything, it is for the company. Point number one, in Bangalore particularly, this part of the world, 10 years ago when we used to speak to people, they would say in your batch record, if there are 3 cuts, it is a typical phrase of Bangalore, 3 cuts, you have to replace the page. If you don't, inspection will fail. The people on the floor would tell this to me, sir, we replaced because there were 3 cuts. So now, this is a matter of, your understanding what the expectations are. As long as what you are doing is right, as long as you can explain what we have done with justification, reason, with notes or with a deviation, everything is okay. So data integrity is an issue of culture and your own understanding of what is expected. So in an attempt to make things look better than what they are, we're getting caught, unfortunately. These are misguided actions, and very often they are quick reactions, sharp reactions, without thinking, done by the people in the midst of the inspection. You understand, in fact, I've spoken a lot in 13, 14, 15 on data integrity, and I did 3 buckets: data integrity during manufacturing, data integrity during inspection, data integrity after inspection. Most of the failures are because of the middle bucket, data integrity during inspection, which is outside the control of management and is not management driven. It is a, what should I call, unqualified, stupid response, reaction given by the people on the spot that can lead to this. So I think we have to understand the whole process and behave in a manner that it is not taken as a data integrity issue. So it is a issue of developing the culture and the practices within your own organization. As far as regulations are concerned, global regulations are available, several guidelines are available. So we just need to follow them and be compliant all the time. And as sir said, it is not the reason, therefore, they are coming for unannounced inspection, right? It is their requirement of the Senate, and it is commonly applicable to everybody. And if that was the case, we would have never found data integrity in the plant inspections, right? We find no data integrity in the plant inspection because the inspectors are very experienced enough to see what happened in last 3 years when you were not there. They have their own ways of digging through things, and they will be able to catch that. So as long as you are doing good work, as per your own procedures, I don't think we should have fear of data integrity. Minus this, unexpected certain quick reactions of making things look better. Just to tell hilariously, I know of a case, sir, that inspector said something is wrong in this file and I need to check tomorrow. And he left the file in the conference room and went home. 2:30 in the morning, 2 people came from QA and QC. They removed the page and replaced it. Real one, I'm telling you. This our name will come, and therefore our jobs will go, reputation will go, and better we try to help ourselves first and then the company. The outcome was definitely, I don't have to tell. So these are the habits and reactions of the people that have caused the impression. Otherwise, as India, as manufacturing, the 4th largest, as sir said, we have to be proud of what we are doing. And this is a passing phase. This will continue. In fact, data integrity, as I've been mentioning, is a lingering issue till as long as humanity remains. It all depends upon within the company what kind of culture you have created and how strongly we work together in not doing these kind of stupid things. Great.
So good evening, sir. Myself, Redder from Symbio Genetics, working in CQA. So my question to Prasant, sir. So regarding process validation. So is there any specific guidelines talks about CPK and limits? And is it necessary to perform the periodical revalidation?
Yeah, good question. The guidance does not talk about the CPK values, but the statistical expert and statistical reference, what you say, it is numerical numbers which are explicitly clear. So what is the significance of a CPK value less than 1.33 is well written, well documented. What is the importance of a parameter or a CQA where the CPK is more than 1.3? The inferences are very clear. So that application should be done as a part of the design of the SOP, and the applicability of these values should be the interpretation after the completion of all the process validation as well as the APQRs.
Right. So maybe I'll take some fillers from the online questions. One is related to what should be the batch size of the lab batch for the stability study for product permission. Because now we have a checklist, but I think Harish possibly it's coming out of your presentation. There's a checklist. You need to give stability data. So what should be the batch size of that lab size batch required for product permission?
As a part of the process validation they will conduct. Okay, so they may be having the process master validation line where they find the batch size. That batch size is based upon the type of the product and the nature of the strength of the product. General batch size, as per even SUPAC guidelines for extending this thing, 1/10 of the commercial batch generally, which is the. But it is just a thumb rule, not exactly. Sometimes based upon your strength of the tablet, other thing, it may vary also. But it is approximately 1/10.
Yeah.
The size of the equipment as
well. Exactly.
Perfect. Any question from the audience further?
This is for Ishwar Reddy, sir. Is there any government-funded labs or place where BAB studies can be done at a subsidized way compared to the private centers, so that will help small-scale industries to go for BAB studies?
I think happy to say that we have been making multiple efforts as a KDPMA, not I'm talking about FOPA, from KDPMA, we have been speaking with the state government for last 2 years. In fact, once the paper also went to the cabinet. Unfortunately, it didn't go through. And in fact, again, in the new industrial policy, they called us last week for consultation. And we have said for a SME, 70% of the cost involved in BAB study should be reimbursed, and for the larger companies, 50%. So the work we are aware of the issues, and we don't want any PLI scheme or a break or anything. Give us subsidy on the BAB studies.
Right.
We have mindset, subsidy, benefit, how much, what is the expensive, what is the cheap. This all depends upon, how you see the thing. Whether 10 lakhs is costlier or 30 lakhs of the BABs, which one is the costlier? So if you feel 10 lakhs itself is the costliest, it is not affordable for the small scale units, we should also see what is the basic expenditure. One part. Second part is that there is a lot of human ethics involved in that. You should see the ethical perspective also. The volunteers is actually in most of the BAB studies, the subjects are the healthy subjects. So you are testing on the healthy subjects. Okay. And as per the even ethical guideline norms, it is the BAB center's responsibility to take care about his well-being and his safety and all the other issues. If something goes wrong to him, he has to pay complete medical management, including the compensation. You may be knowing, recently one South Indian BAB center, they had to pay for one matter 75 lakh rupees to that one particular subject. I see, we have to see other side perspective also. So the number of samples they had to test, these all the bioanalytical methods, and they will use the LCMS and other. So what is the minimum expenditure there? In fact, if you ask the other, US FDA approved facilities fellow, can you do at the 10 lakhs rupees? Over the 10 lakhs mein mera, it is loss for him, he says. So in my opinion, never look at government side for any financial support with respect to that. Yes, wherever, otherwise there is no end for this. Okay, even somebody says that, sir, mera sample HPLC ke liye, I'm sending sample for outside for HPLC testing, he's asking for 1500 rupees for the test. 1500 rupees is very costly for me. You give me subsidy for this 1500 rupees. Oh, there is no any limitation for that. So don't link your quality, your business, your product perspective with the financial perspective, and don't look at the government side. Okay, I understand. You can argue other way, the tax and GST I am paying, income tax I am paying. You name any tax we are paying. It is our money only. Why can't government can support? But I think with respect to the export purpose, I am not sure. Pharmaceutical is doing some support. I don't know, BAB for the registration, overseas registration fee and other thing.
Yes, it is there.
To promote the exports. I think government is, Pharmaceutical is giving some support for the export related. Okay. But this is a good thing, and again, Mr. Harish will send a proposal to the government. It's already gone.
Right. Any question? Yes, many. Yeah.
Hello, sir. Good evening. Myself, Swaminath from CDMX India Pharma. My question is Prashant, sir. Only for my clarification regarding process validation. Sir, even though periodic validation or revalidation, is it required? No significant change has been done for process and system.
Revalidation or a periodic validation is part of the design of the guidance also. Even if you put a frequency 5 years, for example, one variability we spoke about people, you can't demonstrate the variability. You can say the equipment is same, I am doing the preventive maintenance, my materials are same, my vendors are same. People variability is always there. And the assurance on the materials over a period of 5 years by your vendor with whatever controls you have, so cannot be demonstrated scientifically. So looking at all, the frequency has established by the manufacturer. Whenever we say changes, which we spoke about materials, equipment, it's a mandatory, change control required and revalidation required. If even changes are not there for an X period, it could be 5 years, you have to demonstrate a revalidation.
Thank you, sir. And one more question, clarification. What is the exact inter and intra-batch variation?
Yeah. Inter-batch is within the batch. Inter-batch is among the batches. Intra is within the batch. So we spoke about the blend uniformity. So within the batch, for example, even if you say a limit of 90 to 110%, all the 10 samples would pass. The first sample would be at 91%, the last sample would be at 109%. Just to give an example, that variability is already there in the blend uniformity or the content uniformity, what you are talking about. So the intra-batch also through statistical has to be demonstrated for the various sampling locations which you have done. The minimum the variability, the better is the process.
Is it required acceptance criteria for inter and intra-batch variability, sir?
The acceptance criteria for intra-batch is very huge. It's 90 to 110%, for example. It is defined in your process validation protocol. It is a manufacturer's decision, okay, to avoid all the errors in subsequent batches. You can still take batches with a narrow range. This 91%, 109%, which I gave an example, it still meets the requirement as defined in your protocol in the acceptance criteria. But again, the variability is already built into that. But that should be demonstrated in the next 2 batches also. If all the 3 batches variability is there, you can pass the process validation, but with an improvement plan to reduce the variability.
Thank you, sir. One more thing, sir. Good. For the vendor change, if my vendor, alternative vendor, if same process, same ROS, is it required again revalidation?
100% required.
Thank you.
Yeah. Great.
Hello, sir. My name is Silambarasan from Biocon Limited. So my question to the panel is actually, we are now qualifying 2 vendors at it. So whether we need to put 3 batches validation for the stability, or we can reduce 2 batches considering the reducing the stability load.
I think, sir, on a lighter note, we'll have to do one separate program on vendor approval, as it appears. So I'm sure somebody is willing to answer. No, I think getting around the same thing, like, right? Can you repeat the question, please?
Vendor we are validating at the time. So 3 batches we are validated. However, the stability study can be reduced because 6 batches will be validation will come for the stability.
So this was the point I was making in my offline, that if you would like to see what is not written we need not do and how much minimum we can do, then our approach of working together with the systems approach will not work. So it is for you to take a call whether you require or not. How regulation can decide that? Do you get the confidence of the input quality material that is not going to impact your quality attributes of the product? If you want to see, you have to perform. How can we reduce the number because there are 2 APIs? The moment you say 2 APIs, there are 2 different entities coming with a different source, route of synthesis and whatever. In fact, very important thing, what you told to the other gentleman, if the ROS is same, facility same, do you require validation, requalification? It is a challenge to the quality system of the company, not per se to the material. Therefore, it is required. So 2 different entities are 2 different APIs and you require to do whatever you need to do to demonstrate that it doesn't impact your finished product. So I think, not giving direct answer, but the answer is in your own understanding of how it impacts your quality.
I think, in my opinion, it again depends on how whether you can do a risk assessments and what are the risks involved. Whether if there is only there is no major or critical risk, then it's a manufacturer's call. And if you feel that there is a critical risk, then obviously I would suggest, personally I would suggest a minimum of 3 batches stability has to be required. Again, say if you're looking at dissolution as a critical parameter which has to be studied, and if the API is already soluble. So that becomes, the risk is minimum. But if the API is insoluble and even the permeation is a issue, then we are getting into a trouble.
Absolutely. So it is their call.
Yeah,
so I'm very
Just want to add one thing, that I have spent maybe 10 years in risk management. I have not seen a single risk assessment done in order to do more testing, right?
Yesterday,
Always risk assessments are done so as to reduce the testing, right? It's already biased, right?
So
I haven't seen that.
In yesterday Chennai's talk, I gave the formula probability, detectability, severity into hidden A, into red. The attitude with which you are doing risk assessment will determine the outcome.
Yeah.
If outcome is predetermined, your risk numbers will look good to you.
Will be subjective.
PhD
I know.
with the result in mind.
That's what I said. It's predetermined. Then, so I think it is up to you. Through risk assessment is not a permission to avoid compliance either with science or regulation. I think that is the key message.
You had raised a very valid question. So to answer very precisely, in light of WHO guidelines, it is not only the raw metal active moiety vendor change. Wherever the vendor is going to change, you have to take 3 batches. Again, the another step which is written over there, the variability of batch number of API also need to be taken care of. So 3 matrix to be there. And some of the speaker has already mentioned about the person variability. All those are invited the risk. So you have to prove on document that your product is robust, your manufacturing process is robust and producing the consistent quality. Batch number of API, 1, 2, 3 batches of API, 3 production validation batches of these 3 different raw materials, and whenever there is a change of vendor, the another similar set of the process you need to repeat, just like considering it's a new moiety and a new entity. Apart from that, one important thing which was there about the inter and inter, I would like to just say a few words over here. During the protocol designing, there is SD and RSD a specification as an acceptance criteria what the company used to derive. And if anything as per the company norms, it can be further strengthened. As a thumb rule, 3% variability is the acceptance criteria for the process validation as a qualifying criteria. Which is ranging above that is not at all acceptable, and this 3% within the same, like one slide which has been shown as a cone blender, top, middle, bottom, 5 matrix sampling plan, within this sequence the RSD must be less than 3% at any cost. If it is having more, then your process is again having a lot of challenges. Apart from that, from batch to batch, for 3 batches, if the RSD, relative standard deviation, is more than 3, then again your batch is a challenging one. Thank you so much.
Right, sir. I think we almost have completed 1 hour. I really do not know if you would like to continue.
Hello.
My apologies. I had to leave by 5 o'clock, and I have 1 or 2 things to do here. Where is Mr. Melvin?
Sir, can I?
Yeah, we'll allow, yes, of course, 1 or 2 questions. So, yeah, please go ahead in the meantime.
Yeah, myself, Saranya from Syngene International. I have 2 questions. My first question is, for example, can I take a technical batch? Let's say it's a non-GMP batch in a GMP facility. It's like a confirmation batch before proceeding to a GMP batch.
Yeah, non-GMP means? Sometimes, the terminologies, it will be very...
Non-GMP means like I'm not going to analyze all the raw materials or something like that, but I will not be commercializing.
No, without analyzing, without knowing the vendor, without
No.
incoming raw
Vendor...
material, you do the validation.
No, it's like using vendor COA, and we will proceed for engineering batches so that it's a confirmation batch before proceeding to a GMP batch.
As far as my knowledge, engineering batches does not come under the regulatory perspective.
Yes.
That is for you to just decide. That is what I feel. I think he can throw more light on that.
After engineering batch is to be manufactured within the plant.
No, he's saying that non-GMP.
Non-GMP, yes.
That means मेरा घर पे किधर बना रहा हूँ मैं. Somewhere I mean somewhere. No, initially you said
GMP plant.
Is it within GMP or outside?
Madam, I think you said manufacturing non-GMP of the engineering batches or engineering batches in the GMP facility.
Let me come again. It's like a non-GMP batch. Let's say it's a sort of engineering batch. So that I'm manufacturing that batch before I'm proceeding to a GMP batch. It is just for my confirmation.
Non-GMP batch where?
In the GMP facility.
No.
Wow. Then how you can say that it is a GMP facility when you are doing the non-GMP?
No.
The moment you
like,
say that, the moment if you say non-GMP manufacturing in GMP means I switch off the AC, मैं कुछ भी log book पे कुछ नहीं लिखता हूँ, that means I am not following any GMP at all. Actually वो engineering batch, whether it is manufactured or not in that facility also nowhere it is documented. I think आपका question कुछ अलग है. You want to say something but you are saying something.
No, it's not like that, sir. Actually,
Can you give an example and just
for example, I'm taking an engineering batch where
You're talking about tablets or API?
It's a tablet only.
Okay.
So where I'm not analyzing the raw materials from my end, I am relying on the vendor.
I'm sorry to interrupt you. Why you want to make engineering batch?
Just for my confirmation, before proceeding to GMP batch, I want to take this
I mean, you don't have the formulation data, you don't have the formulation.
I have every data. I just want to
If you have
confirm.
every data, then why this engineering batch will come over here?
Because there is a batch size change or there is a client requirement, something like that.
May I help you? I think what she's trying to ask, your question is restricted to whether I can use input materials without testing and by relying on the vendor COE. Is that all?
Yes.
Because now sir is thinking that it will not be recorded in the log books, there is no
it's
history of
not like
manufacture.
that.
So that is breaching the GMP status of the facility.
Non-GMP facility.
Non-GMP?
Non-GMP she is claiming.
No, I think, first of all, I don't like the term non-GMP batch. That itself has to be rephrased.
Okay.
So
It's better to call an engineering batch instead of
yes.
calling it as a
Optimization batch, engineering batch. But anyway, though, to answer the question, if you are taking anything into the GMP approved facility where you are doing commercial manufacturing, no, everything that has to happen there has to happen as per the SOPs. So it means whatever is going, unless you have separate SOPs giving concession or some flexibility in not testing certain things because you are not releasing the batch for commercials.
Yes.
With risk assessment could be a situation. I've seen some companies where they have separate SOP for handling materials to be used for such type of batches within the facility. They will do vendor approval, they will do TSC, BSc risk assessment because you are going to tomorrow contaminate the facility with unknown source of material. All those factors are taken into account. At best, what can be allowed is you may not perform the test and you might rely upon the COE of the vendor.
Yes.
I think that should be acceptable.
Okay.
Yeah.
Okay, sir.
Very quickly, sir,
Sir,
before.
and I have one more question. This will be the last question.
Yeah.
For example, for a product which I'm exporting, can I generate a data, for example, a test analysis with a third party? The third party is not having a license. Is it acceptable? Because I'm not going to commercialize the sample. It is just for my development stage data.
No, where actually you want to export the consignment.
Yes.
Yeah. But, you mean to say that you required any license or some kind of things? It's up to the importing country because here it is in India. If you want to any domestic, if you want to give, then it's a matter of the license and all the things. If it is exporting, then depend upon all importing countries what they required and how their procedure for the accepting that product which is manufactured in your facility.
Rajshekhar sir, I was supposed to come to you with two questions. Since you have not spoken, thanks for taking the mic. Is HEPA filter terminal required for Class D? This is complete out of today's topic, but there are questions from the audience, from the online audience. Is HEPA filter required for Class D? And this is another question related to water. What is the velocity that we need to maintain for flow of water in the loop? You can take or... Or I can deal with the answer, yes. Terminal HEPA filters are recommended.
Terminal HEPA filter is, of course, it is required. Either it is a parental or it is a for solid dosage form. Unless otherwise, if you claim that if the area is a clean room area, then how it is possible without HEPA filter?
HEPA filter, yeah. And so there is a practice that you have HEPA filter in the plenum or you have in the terminal end. But and this is a debate for last decade whether you should need. You may not need for achieving the class. The HEPA filters in the plenum can serve the purpose, but you have issue of contamination of the entire duct back up to the AO. So over a period of time, your entire supply duct will be contaminated. So to prevent that, it is recommended that terminal HEPA filters are placed. In that case, you need not place HEPA filters in the plenum. I think that's the answer.
Maximum, if it is terminal HEPA filter will be there, if my personal experience I can say, and you can reduce maximum contamination and all the things. Yeah.
That's what it is, yeah. I have one small task to do because I need to leave. We have brought from IDMA the coffee table book. Vriddhi is an event for growth IDMA did 3 years ago, Vriddhi 1, and Vriddhi 2 was a mega event of all the associations, all the organizations focusing on topic of growth. So that included domestic market, export market, valuations of the companies, private equities, regulation, of course, USA country director was there. So it covered all types of topics of 2 days at Flame University. And it was a mega event where if you see the book, you will come to know the stalwarts of the industry, right from Dilip Sangvi to many people participated. So to commemorate that and to create a remembrance for that, a coffee table book has been published. And we from IDMA would like to hand it over to you, Sir John Dosage, and the President of KDPMA. Please proceed. So I think so with this, I think we bring the question and answer session to end. If anybody has any other questions, additional questions, you can write and send it to KDPMA as well as IDMA, and we'll definitely get back to you with our answers through either personal mails or through bulletin. So thank you all very much. You have been a wonderful audience. 1 hour where it went, even we didn't realize. Very interesting questions, thought-provoking questions, and I personally enjoyed this kind of interaction. I think this really sums up the way we spend the day, whole day. We are not tired and we still have the same enthusiasm that we had in the morning. Speaks a volume about everybody's interest in the new schedule, and it really augurs well, and we really have made a very good beginning. This is the maiden CDSO and association event that started in Bangalore. So it is a significance for us that we can always claim the first program after the Schedule M was notified, along with the CDSO, happened in Bangalore with a large gathering of 360 people. A really wonderful experience, and I wish really everyone all the best. Thank you very much.
मुद्दा सर ऐसे हम जाने नहीं देंगे आपको. Thank you, SM Muda ji, for conducting such a lively question and answer session, and as a certificate of appreciation, and I request Dr. Ishwar Reddy ji to kindly hand over a certificate of appreciation to Muda sir. Now moving ahead in the agenda, I request Shri Sunil Attawar past President KDPMA and CMD Group Pharmaceuticals, to give his concluding remarks and vote of thanks.
Thank you, Harish. I'm sure many of you follow cricket. In 2014, you may remember that the IPL was shifted from India to South Africa because of we had the elections. And at that time, somebody asked Harsha Bogle, how did you all manage this? In just a matter of some short time, you shifted the entire IPL from India to the entire whole thing there and made it a great success. So his answer was, in India, nothing happens till 4 weeks before an event. And in 4 weeks before the event, everything happens. The Schedule M draft came in 2018, if I'm right. It was in this hall in 13th October that, in last year that we had met before the Schedule M, and the discussion was whether it's going to be postponed. And I think most of the questions was that. Today, the hall is full. You have sat down right up to this time, and I think that's a testimony or a fear that Schedule M is at the doorstep. For companies above 250 crores, you have just few weeks. For those below 250 crores, I think you have 6 months. So I think we are all prepared, and like everything that we do, the last leg is going to be very, very significant for us. And I thank CDSCO, IDMA, KDPMA, and the drug controllers for actually having this program. And the topic is very relevant on product and process knowledge. I think right through the day, what we have seen is that the basis of good GMP, the basis of good products, starts at the design stage. It starts at that stage, and I think it is a fact. If we are giving 6,000 licenses a day or even in a month, the fact is that a lot of work is not happening on the design and the process. And I will give you my own example. We had a factory in Mumbai, and we shifted that particular product to our Bangalore unit. That was when the first Schedule M came, and the factory in Mumbai was not meeting the standards. And everything is the same, transfer knowledge. Even the manager and the supervisors were the same. But when we started the validation batches, the batches were failing. One batch, second batch, failing because the product was getting hard in the tank. We make semi-solids and was getting hard in the tank. We tried everything, but it was still failing. And the answer used to always be human error. Maybe the workers are not doing the right thing or something like that. Till we did a detailed RCA, and we found out ambient temperature in Mumbai is always above 35. In Bangalore, ambient temperature in December is below 22. So because you're cooling the tank at different temperatures, the process is like a shock cooling that you are getting, and it was impacting the batch. So a small thing like that also can impact a process, and if you do a good RCA. So I think right in the morning, Mr. Mudda started by saying that 94% of failures are due to process, just 6% due to people. But most RCAs, and because I see in our own factory and otherwise, is where we attribute every failure to human error. In fact, we work for a lot of companies. There was a multinational we worked for in which it was a rule that you could not put human error in your RCA, and you could not put training in your CAPA. It was a rule, and we had to follow that rule till, even now, that you can't put human error or you cannot put training, because if your training is faulty, then there is another problem which you need to address. And that is why today's program on process and product knowledge, I think, was brilliant. It's a great start, and I think all of you, the fact that you're sitting here, you believe that this is something that is very important. I think Schedule M is something that is only going to bring good to the industry. It is going to bring a lot of more business. People are worried that business will go, but I think business will increase, because we all know in this industry, reputation is what you have, is the only thing we have actually. And when reputation increases, your price will increase, orders will be more, the whole world will look at India not as a rest of world supplier of medicines or a generic supplier, but they will come to us to get the best quality medicines. So if I have to sum up, I'll just take one point from each speaker. Of course, Dr. Ishwar Reddy started, and from a regulator's point of view, and Schedule M, if you see the difference between the old Schedule M and the new Schedule M, what has happened is the old Schedule M talked a lot about the spirit of GMP. And in India, unfortunately, we don't follow the spirit of law. We always follow the letter of law. So when the spirit of law is there, and we find the easiest deviations, when the letter of law comes, we are enforced and we are forced. Nothing has changed between the old Schedule M and the new Schedule M. It's only the spirit has been changed to letter. Everything that is there is now very specific. Product and process development has become mandatory. Even earlier it was there. It was not, nobody in old Schedule M said that you can start a product without doing stability. I think it was already there. But now it has become mandatory. It's very important, and I think Dr. Ishwar Reddy has been very clear in that, and Schedule M is also very clear in that. And one point that he put, and he said, participation, support, and commitment of staff in all levels is critical. Everybody blames everybody in the company. The juniors blame the management blame the juniors, and it just, it's something that we must all realize that quality is not the domain of one particular. Management sets the culture, absolutely. If the management has a bad culture, it will go down the road. The supervisors also need to follow process. The juniors, everybody needs to follow process, then only things will improve. And I think what Sir said, that participation, support, and commitment of staff at all levels is become now letter of the law in new GMP. Harish, of course, in his usual style, has explained the entire process of product development, and what I will pick up from his speech was that product development would start by design. We should know why we are in the market, why we are developing this product, what is the end expectation, what am I comparing it to. Only then we should initiate the process. Of course, Prashant took us to the complete process of validation, and how to build a robust process in development. And Mr. Vyas, he said one thing which I liked very much. He said, make compliance easy. And I think all of us sitting here are in some role that we play on the shop floor. If we make compliance easy, everybody will find it easy to follow. Like you gave an example that how do you expect people to stop at the signal when the temperature is 45 degrees? So if you put sprinklers, people will stop. You know what they did in Bangalore? They put dummies of policemen. Because we are so afraid. So for a long time, at every junction, because people will break the signal, and they put dummies of policemen, so the person will see, oh, there's a policeman, and he will not clean. So that is how we are. That is our culture. That is how Indian we are. If nobody is watching, we will do, something. But we all know GMP is not that. GMP is to do, when nobody is watching, you follow process. That is very important. So I'd like to thank all the dignitaries on the dais, Dr. Ishwar Reddy, Mr. Mudda, Mr. Naresh, Harish Jain, Mr. Prashant, Mr. Vyas, for their wonderful lectures, and also great participation in the panel discussion. I think a lot of answers were answered, but I still a lot of questions that you have. Thank you very much for being a very important part of this program. I'd like to thank Dr. Khanapure and the team at the Drug Control for giving us their continuous support. I'd also like to thank IDMA, Mr. Mood is there representing Mr. Viranchi Shah, is also always talking about GMP. I don't know whether there's a change in leadership now, but I think he's there. Our president of KDPMA and the entire team at KDPMA for bringing this program. Two important, I think we have to thank the sponsors, Microlabs, Parley Global, and if I'm not wrong, SVN Technologies. technologies for supporting this program, because of which we could subsidize it, substantially subsidize it. So if you take, I think it's almost free for most of us that are participated in this program. I also like to thank all the support staff, photographers. We had the press in the morning, our staff at KDPMA, including Mr. Shastri and team. And of course, to thank all of you. You've been a great audience. I think a 4 days program you sat down through this whole program, which is rare. Usually after 2:30, at least we see a lot of vacant seats, but you've been a wonderful program. Just one takeaway for you, which I have, and a request, when you go back, at least please share this with at least one other person in your faculty. I think that will build also a culture that wherever you went, you learned something. Whatever you learned, it doesn't have to be what I said or what the participants said. Whatever you learned through the program, you just pick up one colleague in your facilities and share what you learned with them. I think that will bring up a culture of quality. Thank you very much for this wonderful day.
Thank you, audience. With that, we are coming to the end of the programs today. So tea has been organized on the 4th floor. So please have your tea and coffee before second floor. So you don't have to go to the 4th floor. The tea is there, coffee on the 2nd floor. So please have tea and coffee and thank you very much. I hope to see you again.